Technical team, am I audible? Hello, am I audible? Manoj sir?
Yes, sir. Vaibhav, sir. You are audible.
Thank you.
I think technical team should share YouTube link on the group that the remaining participant can join from YouTube also.
Meeting is now streaming on live on YouTube. I think we shall wait for a couple of minutes. Let the
Yeah,
participant number
I have,
increase.
I have just received a call from Dr. Singh. He has informed me that he is
Yes, sir.
in the process of joining.
Right, sir.
नहीं करते हैं मुलायम है तो
Is Dr. Basarkar sir online?
Yes, sir. Yes, sir. Good morning.
Yes.
I think there must be a waiting room created, and Dr. Singh is waiting for him to join.
Hey, Dr. Singh has joined. Good morning, sir. Namaste.
Very good morning.
Namaste, sir.
Good morning, sir.
Good morning, ji. Good morning to all.
Good morning, Singh, sir. Dr. Basarkar here.
Yeah, how are you? Good morning.
I am fine here. Hope you are well, sir.
Yeah, it's all fine.
It's all fine.
All fine. Yes.
We are happy to see you here. Happy.
Always with you. Always with you.
Thank you, sir.
Right. Just I have to go daily for dressing after
Oh.
the surgery.
Oh,
So I will go in the afternoon today.
Much. I hope it must be fine now.
Yeah, it is getting better.
Okay.
It was spacious cyst that got inflamed. So
Hmm.
they had to do a surgery to remove it.
I request all the participants to just mute their microphones. Some of the time it may create some confusions. Thank you.
हाँ ना यार मैडम क्या पता तेरा सब का मैंने ज्वाइन कर
Okay, I think Sunil Babu has also joined. He is traveling to some place.
It's about 45 participants joined now. If the chair permits, we can commence with the program.
Yes, what I feel, our technical team should uh, keep 10 connections reserved and allow everyone to join. Yunus, I think up to 90 you can allow everyone.
Okay, sir.
Okay, and keep 10 connections spare
Reserve.
for management and other respected dignitaries.
Yes, sir.
And on YouTube also, around 16 people, 20 people have joined. So let us begin. Vaibhav, please.
We'll begin.
Yes, sir.
That is already 10:51.
Yes, please.
Yes. Vaibhav sir,
Okay,
please
right.
proceed.
Technical, yes. Technical team is requested to share the screens. Durgesh sir, we need to observe the backdrops. Once I get a green signal from the technical team, I'll surely start. Can you put it into the slide mode?
Let's go down.
कन्नौज का वो अत्तर साज नहीं. With the permission from the chair, I'll be starting with the program. Thank you, technical team, for the good presentation. Good morning and a warm welcome to all. At the outset, I extend the most respectful and cordial welcome to our esteemed trust members, honorable dignitaries, our distinguished resource persons, and all respected academicians and delegates, researchers, and participants joining us today from various parts of the country. On behalf of Academic Trust, it is my proud privilege to inaugurate the National Conference on Annual Academic Endeavor that continues to serve as a meaningful platform for knowledge sharing, intellectual dialogue, and technical advancements. Over the years, this program has grown in relevance and reach, reflecting our collective commitment to academic excellence and national development. Though we are gathered on a virtual platform today, the spirit of tradition and reverence remains undiminished. Before we proceed, we humbly seek divine blessings by invoking Lord Ganesh, the remover of obstacles, and Goddess Saraswati, the source of knowledge and wisdom. And as a mark of our deep gratitude and remembrance of our revered founder, late Karmavir Keshavlal Jiabad, fondly called as Kakaji, whose legacy lives with vision, values, and dedication, continue to guide us and inspire us. I now request you all to join me in thought and solemn reflection as we symbolically light the ceremonial lamp, invoking wisdom, clarity, enlightenment, and purposeful learning. Thank you, technical team, for the parallel presentation. With this moment of remembrance and renewed purpose, we now formally inaugurate the national conference entitled as SNJB Pharmacon. SNJB stands for Sri Neminath Jain Brahmacharya Ashram as our trust, pharmacy as our background, and con represents conference. It is SNJB Pharmacon 25, and the theme sets in as practical and regulatory considerations for stability indicating methods in assess. I'm confident that the deliberations, discussions, and outcomes of this conference will contribute meaningfully to academic growth, technical innovation, and national progress. I wish the conference every success and hope it proves to be intellectually enriching and professionally rewarding for all of us. At this juncture, we proceed to the next segment of preamble, which will be led by Dr. Ganesh Basarkar, coordinator of SNJP Pharmacon. Over to you, Dr. Ganesh Basarkar.
Thank you very much, Mr. Vaibhav Buchke, sir. Good morning to all distinguished guests, respected academicians, researchers, industry professionals, faculty members, and dear students. It gives me immense pleasure to present the preamble of SNJB Pharmacon 25, a national conference organized by SNJB's College of Pharmacy Chandwad, with the objective of fostering academic excellence, research culture, and meaningful interaction among the academicians, researchers, and industry professionals in the field of the pharmaceutical sciences. This prestigious academic event is being organized under the guidance and visionary leadership of our esteemed chief patrons and patrons. I express my deep sense of gratitude to Honorable Sri Babylalji Sancheti, Chairman SNJB Trust Board, Honorable Vice Chairman Sri Dinesh Kumarji Loda, and Sri Ajit Kumarji Surana, Chairman SNJB Prabandha Samiti, and Sri Arvindji Bansali, Vice Chairman SNJB Prabandha Samiti. At the same time, I express my sincere gratitude to our Chief Coordinator, Sri Jumbarlalji Bhandari, and Coordinator Sri Sunil Kumarji Chopra, Advocate Sri Prakash Chandri Boktia, Sri Yogeshji Modi, Sri Abhijitji Modi, and Sri Anandji Chopra for their invaluable guidance, constant encouragement, and inspiring leadership and support towards the promoting quality education and research initiatives. I deeply honored to have Dr. Saranjit Singh, former director, NIPER, Chandigarh, a renowned academician who is mentor for us since the starting of the PharmaCon 2017. His vast experience, scholarly contributions, and visionary guidance have been a great source of inspiration in shaping the theme, academic rigor, and overall structure of our every conference of the PharmaCon every year. My sincere gratitude to Sri Pramodji Galankar, Administrative Officer, for his valuable guidance, administrative support, and wholehearted cooperation. My sincere acknowledge the dynamic leadership of Dr. Chandrashekhar Upasani sir, Principal SNJB College of Pharmacy, whose continuous support and academic vision which have been instrumental in the successful of planning and execution of every year's SNJB PharmaCon. Dear friends, SNJB PharmaCon is a flagship national conference. It is a series initiated by Suresh Dada Jain College of Pharmacy, Chandwad, in 2017 under the aegis of Sri Neminath Jain Brahmacharya Ashram, with the objective of promoting academic excellence, research culture, and professional interaction in the field of pharmaceutical sciences. SNJP PharmaCon has a rich and inspiring journey. What began as a simple, modest academic initiative several years ago in 2017 with a vision of creating a platform for knowledge exchange, now it has grown into one of the most respected academic conferences organized by our institution. Over the years, SNJB PharmaCon has evolved into a prestigious academic event, witnessing enthusiastic participation from reputed institutions, research organizations, regulatory bodies, and pharmaceutical industries. Each edition of this conference have been thoughtfully designed under the leadership of Dr. Saranjit Singh, with the contemporary themes covering diverse domains such as pharmaceutics, pharmaceutical chemistry, pharmacology, pharmacognosy, clinical pharmacy, regulatory affairs, and emerging interdisciplinary areas. This year's SNJB PharmaCon holds special significance with the theme of practical and regulatory considerations for stability indicating methods in essay, which we have considered, which may be useful for all the faculty members as well as the industry people. As we step into this 9th edition, we celebrate not only just a milestone of the year, but a tradition of excellence built over the years of dedicated efforts by all our faculty members, leadership, and our enthusiastic participants. This conference aims to highlight contemporary trends, emerging technologies, regulatory transformations, and innovation reshaping the pharmaceutical sector. As we commence this year's conference, I invite all the participants to actively engage, exchange their insights, share your experiences, and contribute to the collective knowledge building that SNJB PharmaCon stands for. Let us celebrate the legacy of our past, embrace the opportunities of the present, and future work together to build a stronger, research-driven future for the pharmaceutical community. I am confident that SNJB Pharmacon 25 will be an enriching and rewarding experience for all the participants and will contribute meaningfully to the advancement of the pharmaceutical sciences. I'm also grateful to our advisory committee members for their constant guidance, encouragement, and support, which have been instrumental in shaping SNJB Pharmacon 25. The wholehearted involvement of faculty members, supporting staff, organizing committee, reviewers, and student volunteers has made this academic endeavor possible. Let SNJB Pharmacon 25 inspires innovation, collaboration, and excellence in the pharmaceutical sciences. Once again, thank you, and I wish SNJB Pharmacon 25 with a great success. With due respect, now I would like to invite Dr. Chandrashekhar Upasani sir, our principal sir, to kindly deliver the welcome address at the inaugural function of this session, SNJB Pharmacon 25. Thank you once again, and over to Dr. Chandrashekhar Upasani sir.
Thank you, Dr. Basarkar. At the outset, I welcome all the dignitaries and especially our brand ambassador, Dr. Saranjit Singh sir, for hosting this SNJB Pharmacon 25. This is a silver jubilee year of our college, and at this silver jubilee year of our college, Dr. Saranjit Singh sir has given us a theme of practical and regulatory considerations for stability indicating methods in assays. What I feel is the theme of Pharmacon is a very important thing, and it should be igniting the thought process of all the technocrats. And with this view, we are working on SNJB Pharmacon every year. This year, last year we could do it physically, but this year Dr. Saranjit Singh sir was not so well, so he requested us to be present, to remain present on online mode. So we thought that the conference should be made on the online mode. So with this note, I welcome Dr. Saranjit Singh sir, who is a mentor for this SNJB Pharmacon for so many years. And I welcome, I could see here Honorable Shri Sunil Kumarji Chopra, who is a management member of SNJB, and his presence is also here. And at the backdrop, I could see the other management members here. So I welcome one and all the delegates, all the resource persons, and all the participants for this SNJB Pharmacon 2025. Without wasting much of time, I would like to start the proceedings, and I request the technical team to start it. Thank you.
Thank you, sir. Good morning, everybody. I believe I am audible enough. Due respect to honorable coordinators of SNJB's SSDJ College of Pharmacy, respected principal, coordinator of this event, SNJB Pharmacon 25, and my dear delegates and students. Friends, I derive immense pleasure, immense pride in introducing Dr. Saranjit Singh sir. Guruji has led an illustrious career. He is a shining alumnus of UDPS, Punjab University, Chandigarh. He pursued his PG in pharmaceutics and has been winner of a gold medal at MPharm. He then started his career in pharmaceutical analysis at NIPER, that is National Institute of Pharmaceutical Education and has been a former dean there. This NIPER Chandigarh has been the premier establishment of Government of India catering in pharmaceutical needs, pharmaceutical scientific needs throughout the world. Dr. Saranjit Singh's efforts and talent has fetched him many awards. Professor M.L. Khurana Lecture Series Award is one of them. His endeavors in SIAM, that is Stability Indicating Analytical Methods, have been seminal in changing the perspectives of whole pharma world towards the issue of stability and expiry of formulations. His findings has left a profound mark and impact over the regulatory authorities and has been immensely helped in framing the guidelines, several guidelines concerned with generation and analysis of stability samples. Guruji has been very benevolent and has been associated with SNJB since 2017. This is his this is the ninth edition, and he has been going strong. His constant persuasion, his nobility, and encouragement has been profound in transferring his experience and knowledge to the young generation. This is the ninth edition, as I said, and he has been bringing the same experience for the younger generation. In his last visit, a prolific speaker he has been, in his last visit, he has mentioned that that was his 601st lecture which he has delivered at our campus. This year, he must have added many more. Let us now all welcome Dr. Saranjit Singh over mic and witness his expertise, which will definitely be enlightening us and helping us in our future endeavors. Over to you, sir.
Thank you so much, Dr. Vivek, for such a nice introduction.
Thank you, sir.
I'm extremely happy to be there at the inauguration of the ninth edition of SNJB Pharmacon. It is my honor and it's an esteemed honor being provided to me that I have been given the role of being the chief guest at every SNJB. And first time I thought that I should request Dr. Upasani that let us have something to discuss upon the area of larger area of stability testing and smaller area of stability indicating methods. And he agreed last time that sure, next SNJB will focus on these issues. In the previous editions, they were all variety of topics and very different topics in all 8th editions. So I'm very happy to be back here. I'm sorry that due to health reason that I could not physically travel. And because the IPC is also very near, so it was collectively thought of that we should have SNJB 2025 online this year, and which was also agreed upon, and today we are into it. First of all, to the whole, my first respects are to Kakaji, the great personality who mooted the whole education in the area of Chandwar, and the college was set up under his patronage. And I'm very happy to really congratulate the whole management, past, current, and future managements, the team members of the college, and everybody on your silver jubilee this year. I am sorry that I could have enjoyed my physical presence on the campus, which is a beautiful campus. and interact with the students face to face and the faculty face to face. And I had always enjoyed practically the full house during the sessions of SNJB, and very rightly said that SNJB Pharmacon has the spectrum has enlarged over the years, and it has become a very prestigious conference over the years. So I am happy that Dr. Upasani's leadership is really making wonders to the college. And the college at this remote area of Chandwad, 65 to 70 kilometers away from Nasik, is really in the limelight now. And I have interacted with many of the speakers, previous speakers of the Pharmacon. They have really had a great enthusiasm to be present and come to SNJP Pharmacon every year. And this is what the magic is happening over the years. And I have all the best wishes for future also. And I promise that as long as you wish, I will be always there to be with you at the Pharmacon conference, this conference. Now, good morning to all the participants, everyone. And the being the inaugural discussion that I want to do with you, I will share the screen now. Just let me open the presentation. Now, is it visible or yet or not?
Not yet, sir.
Okay. Just let me go to Zoom meeting. Now, friends,
Yes, sir.
is it
Yes,
visible?
sir.
Very
Very
much.
good.
Fine, good. I think I have to go to the speaker view. I myself cannot view yet the screen.
Okay.
Let me see. Somehow I cannot see the screen, my presentation.
Hello. May I ask the technical team to assist?
Now, is it visible, the first slide?
Yes, sir. It is visible.
Okay.
It is
So,
visible, sir.
okay, good. Now, what my plan had been that because stability indicating assays are essentially a part of the whole stability testing program in industry. And that was a reason that I thought that I would start with stability testing as such a large level, broader level, and talk of the importance and regulatory consideration within the realm of stability testing, and talk about stability indicating assays also as a part of the whole stability testing program. And then other speakers who are from industry and academia who had been actively working on stability indicating assays and method validation will be talking about, and leaving that part to more detailed discussions part to the other speakers. So broadly, I will talk on stability testing, why we do stability testing, what is the importance, objectives, and then what are the regulatory guidelines under which we are directed to carry out stability testing program and uh, conduct the stability indicator, develop and validate the stability indicating methods for getting the shelf life estimates. So all of you are aware that whether it is Indian pharmaceutical industry or global pharmaceutical industry, all over the world, there is intense regulatory scrutiny happening. Today, the FDA inspectors or inspectors from any of the importing country will come unannounced to an industry. And whenever we had tried earlier also for SNJB Pharmacon to have industry speakers, some of times, many of them back out because they say that our audit is going on. And this audit is unannounced. So almost it's a perpetual audits that are happening. And everything you do in industry, or everything you document is looked into very deeply. And the focus on stability testing and stability indicator assay is also very critically there. And the reason is that basically the premise is that every patient has justified reason to receive drugs that have optimum quality, safety, and efficacy. And believe me, whenever now that I am into some small health issue and I have to take a lot of medicines, and first thing as a customer, as a patient, what we do and look into whether the drug is expired or not. I have been into stability testing field for now almost 45 years, and I know which drug is more stable, which is less stable, and accordingly, sometimes I could take a risk to take the drug within 1 month of the expiry date if it is lying at home. My wife always will warn me before taking the medicine. You first check the expiry date because you are a little, as an expert, you take advantage of many things, but be sure that it is not surpassed the expiry date. Now, sometimes I laugh at it that because I'm a stability testing person, when I used to be in NIPER, that I will take some advantage and leverage that I will allow drugs to expire at home, like you people also. I don't know how much critically you look into your box, the first aid and drug box. And in my home, I seldom use allopathic medicine. Rarely I use, other than 1 or 2 routine medicines for cholesterol or prostate. Otherwise, I don't take a lot, means the allopathic medicine that often, and most of the drugs will expire at home. So that's why my wife knows that we have a lot of expired medicines in our drug box. So she always warns me that make sure that drugs are not expired before you consume it. Rather, I used to laugh that I always allow them to expire to test them in my lab and develop stability indicating methods on them. So that was our discussion that always we used to have. So basically, essentially, that whenever the patient has to take a medicine, it has to have an optimum quality, safety, and efficacy. And I am talking of one of the lecture I delivered at IDMA Pharmaceutical Analytical Convention, Analyst Convention, long back in 19th, that one. And if you see at the back, the banner, it said patient safety first. So this is in focus of all the organizations. Sometimes some of the industrial houses who ignore this when they don't put the patient safety first, that leads to the problem that we are facing now of all the cough syrups and governments, again, Indian government regulations and strictness over the manufacturing houses and Schedule M application, the new Schedule M application, which has come must for every small or medium or large industrial houses. And believe me, I have not including the slides somehow. And there is a much, much greater emphasis on stability testing in the revised tool M, latest tool M. And if you look into detail discussion of revised tool M, there is a lot of places where the stability testing now is having a greater emphasis. So the other reason is that products are manufactured by pharmaceutical industry in very huge scales. Believe me, the modern tableting machines, uh, you may be able to imagine, I don't know, yes or no, the speed of the one of the latest tableting machine is 5.5 lakhs per hour. And you can think over whenever a batch is produced on such a high speed machine, how many tablets would be produced in a day or in a month or in a years? Maybe millions of pieces. So very rationally that because these products are manufactured in such a large scales, and even a single batch failure can affect such a large number of patients who consume these medicines. So that is the reason, primary reason, that the regulations are implemented stringently and they are to be respected by the industry. Now, the emerging emphasis, global emphasis, is one drug, one quality. It is required that the patient is same everywhere in the world, whether it is in Chandwad, Nasik, Chandigarh, Mohali, or anywhere else in the world, any country, anywhere. There should be one quality for any molecule, any formulation. If it's a tablet or let us say paracetamol tablet, it should be same quality everywhere in the world. That's why you know that all these ICH came into picture and harmonization of regulations so that there is one quality that is produced. Somehow, unfortunate part in India is that our industry is highly fragmented. We have multinationals, Indian multinationals, large companies, medium companies, small companies, and to for anyone, even regulatory system, to ensure that it is the same quality everywhere by manufactured by every manufacturer. This is the goal of what we say is the revised ULAM or otherwise. And that is the reason even the foreign inspectors, when they come for auditing of manufacturing plants, today they will also enter your washrooms to see what is the level of hygiene in your washrooms. Because they say that if it in the R&D center, the washroom is dirty, and that culture of low hygiene will also be there in the manufacturing plant. So they will even go to the R&D center. They will go to every washroom in the facility, whether it is a production or R&D or your quality assurance or whatever department associated. So this is where it is a very difficult task for the world agencies, organizations to make sure that the quality is same. Basically, we are in the process of harmonization of pharmacopoeias, ICHQ4, and there's a pharmacopoeia discussion group which is doing that. But yet, only some of the general methods for testing of the drugs have been harmonized, like dissolution test, disintegration test, that kind. They are harmonized all over the world. Even Indian pharmacopoeia is following the same, but none of the monograph has been harmonized yet due to cultural differences in different parts of the world. So until unless the monographs are also harmonized to make sure that there is a one drug, one quality, because the monograph specifications for every drug and every product need to be harmonized world over. So USP had made an effort that they have USP of the one, and they also came out with the monographs for international use. and having the same standards as USP. Because USP essentially is meant for being followed in US or any product which is labeled as USP world over. But unfortunately, what happens was that they wanted to impose the international standards which they developed, which were parallel to USP standards. But that is the molecule or the product becomes costly exercise for patients if you follow the most stringent pharmacopoeia. So different pharmacopoeias in the world, not all countries have their pharmacopoeia, they follow one another. Indian pharmacopoeia now has improved its specifications in monographs, and by that virtue, it has been adopted now formally. MOUs have been signed with 18 countries, but I will say these 18 countries are still less regulated countries, which are giving partnership with the Indian Pharmacopoeia Commission and acceptance of the standards of Indian Pharmacopoeia. We are copying monographs from BP and USP uh, under, again, MOUs that we can take up the monograph wherever required from and follow up from the pharmacopoeia, other pharmacopoeias like BP, EP, USP, trying to harmonize as much as possible so that we also have claim of that we are also following the global specifications within the pharmacopoeias. So friends, this is a very big challenge. And because we have to export the products out, and the products manufactured in India, if they have to reach US shores or any other country which is far away, thousands of kilometers, maybe 5,000, 10,000 kilometers from India, and products reaching in stable form over there in those countries is another big challenge, which is a shipping stability challenge. So these things you have to keep in mind, that the world is trying to have same level of specification and standards in testing of pharmaceuticals through pharmacopoeia or regulations which are imposed on the rest of the world. So when the same specification standards and regulatory requirements are not met, the regulatory agencies issue warning letters, and many of the products currently are being recalled due to stability failures. I will give you some of the examples over here. Like FDA 483 is a warning letter concerning stability testing. So there is a full article which was issued somewhere on in November 2023, and they have really discussed what are the problems in stability testing that the world companies really are facing, and they have real problems, and that's why warning letters have been issued. Again, FDA warning letter highlights, this was of October 2025, 8th October, happens to be my birthday on that day, 8th October, but on my birthday, even the FDA is issuing warning letters on OS handling and stability failures. Top compliance issues in the pharmaceutical industry, FDA 483 reasons and examples in which there is again clear mention on stability testing failures. So I continue looking into the publication. This was on August 1, 2025, and what type of problems industry facing with respect to stability testing failures. This is an interesting reading for anybody. What are the basically reasons and the examples they give. This here they say that common findings included a previous para it says here a detailed analysis of financial year 202 form 483 inspectional observation revealed that deficient stability programs governed by 21 CFR 211.166 contributed to a significant compliance gap. So common findings included no written stability testing program, stability programs are not followed consistently, invalid test methods, inadequate sample storage, design, or missing long-term data. So invalid test methods, which will be the main theme of this conference. So this is one of the key areas where failures do happen, and people have not understood how to develop the validated test methods. Uh, then Glenmark Pharmaceuticals Limited got the warning letter on July 11, 2025, and it's a your firm failed to follow an adequate return state testing program designed to assess the stability characteristics of drug products. Now, whole of the discussion in their warning letters focus on stability. Now, the problem here is in industry that whenever you are a fresher passing out from a college and you join a company, you'll be put into the stability testing facility. The reason is the Indian industry doesn't understand real the importance of stability testing, and they will put freshers in that area, and there will be because freshers are trainees, and they will tend to make mistakes. Industry has not really understood the importance of stability testing and systematic, uh, practically the importance of the stability testing. So that why there are less of trained person. They put freshers in the stability testing area. So that's why these warning letters are being issued. And then product recalls are happening also world over. So you can see over in Kenya how many of the products were. You can see over here color change of the product in to black, color change in the product to black. There, this was a product from India. Again, a product from India. Again, third same company, Kamala Amrut Pharmaceuticals. Color change in the product to black. And these are all paracetamol products of different strengths. Failure to comply with assay identified during long-term stability study. Failure to comply with the release assay. This was Kenyan product. Color change of the tablets. So question is why these things are happening? Hmm. Certainly because there were not. Now Kenya is a less regulated country, but now they have fallen into the because of the insistence of the global agencies WHO that you develop your regulatory system, and now they are also putting such list. Africa being an African country, still they are putting such a list, and they really this is leading to the product recalls in even less regulated countries. This is a CDSU alert list, and you can look into multiple products and content of ampicillin, amoxicillin, tridate, and cloxacillin sodium. So there is issue over here, lower content. It means the product has shown stability, instability over the period, or it can be intentional low content, but maybe stability reason. Dissolution of sulfurs, methoxazole, content of ambroxol hydrochloride, content of salbutamol in salbutamol sulfate and ambroxol hydrochloride, pH and content of azithromycin. So this way, the content of zinc oxide or other things. So some of the drugs which are unstable will try to fall on stability, and if it goes beyond the pharmacopoeia limits, so the product is declared at NSQ, not of standard quality. So this is June 2025 NSQ from the state, and these are all tested in different states labs. So Sun Pharma recalls over 5,000 bottles of ADHD drug in US. The reason for the voluntary recall is stated to be an out of specification results observed in dissolution test during analysis at 9-month long-term stability station. So how the product will be stable for 2 years if there is a dissolution failure at 9 months? So they are very, very, and if such recalls happens and warning letters are issued, immediately the share of the company goes down. So this has to be viewed very seriously by the companies. So Cipla recalls over 20,000 packs of asthma medicine albuterol in US. So again, this is a particle size in the induction port exceeded the specified range. The voluntary recall was ordered after the drug failed stability specification at a 12 months check. Particle size changing. DRL, Zydus recall drugs in US over manufacturing issues. So here also there are stability testing failures, injections over stability testing failures, while Zydus Life Sciences had a different reason. So basically, almost this is very routine that stability testing and stability result failures are being shown by products manufactured by industry, and it's not an Indian phenomena alone, but it's world over. But there are quite a good percentage of Indian products failing. So we all, whether the students, faculty members, have to really focus on stability testing, the appropriate stability testing in industry, so that we can train our students in this field. And whenever they are joining and they are put freshly into the stability testing facilities, so they are pre-knowing what are the problems that our products can face. So declared reasons of recalls due to product stability issues are as follows. Failed degradation product specification. So degradation product has gone high. That's why the product was asked to be recalled. Failure in dissolution, change in physical practice like discoloration, precipitation, generation of odor, etc. Incorrect expiry date, low potency, etc., etc. So there are multiple reasons that have been cited in the warning letters and the recall reasons that due to instability of the products due to multiple reasons. Now, one thing which we have to keep in mind always, that we are manufacturing the product under very ideal conditions. If you go to the pharma company today, not even a speck of small dust is allowed inside the pharma manufacturing facility. You have HEPA filters, control temperature, control humidity, everything is ideal. But unfortunately, the moment the product is manufactured, packaged, and into secondary or tertiary pack, and what happens? It lands up into a non-ideal situation post to manufacturing. Now, this is again a very big issue that is not being taken care well by our manufacturers. So we have ideal production environment. There are whole regulations and controls. We have good manufacturing practices, good laboratory practices, and ours. You can see the manufacturing area is so clean, dust-free, all coving on the ceiling and the floor bottom edges, and where you can see the shining area, almost 6-7 star type of facilities. So we get an ideal formulation. Post to manufacturer, there is a truck standing outside and non-ideal shipment and storage environment. Now the question is, it has happened, it can happen at transport level, wholesalers, retailers. Now you say you store at temperature of 2 to 8 degree. Okay, protect it from not more than 30 degrees centigrade. So which is a normal country's average temperature taken. So we say do not store above 30 degrees centigrade. So question is whether the truckers, when it is being transported in rail or by air, now you can see the products lying on in the sun. So all these, when the product is being distributed, shipped to anywhere in the world, are the conditions which are required in which you have tested that the product remains stable, are being stringently followed at the transport level, wholesalers, retailer, hospital stores, or even patients. Do you at home read the storage condition on the product and accordingly store your pharmaceutical product which you are consuming, and make sure if it is stored below 30 degrees centigrade and within a certain low humidity condition, and you are not putting the product into your washroom, and to keep it away from children, it is put into the washrooms at times. So question is, it's a high humidity area. So whether at patient level also, I tell you, there is less amount of drugs today in the chemist shops or the wholesalers. Most of the products which are unused is lying at our homes because we don't follow prescription exactly. We don't take medicines regularly. We skip doses. And after, because the pharmacists will give you earlier the cut strips are allowed. Now they will say no, we will don't cut the strip. Government has said no, you cannot cut the strip. You need 4 tablets. They give you 10 and 6 are lying with you. And it may be lying with you until unless you open the storage box and expel out all those products which have expired. So the longevity of the product in chemist shop, it is very low because people are coming and purchasing and the fresh stock is coming. But the longevity of the products at households is much, much, much longer. And huge quantities of products are lying at households. So we are responsible for mishandling. We are what we are doing. We have a drug box. We remove the products from the drug box, take that tablet out or any unit dose out, keep it back. Next time we are again shuffling the blisters or strips which are lying multiple one in the drug box, and we are allowing it to scrap over each other and thinning off the packaging materials. So everywhere you will find that manufacture is okay. They have manufactured the product under ideal condition, but it is mishandled at all levels. Now, interesting case is one of the case which went to National Human Rights Commission also was of Gujarat company co-parenterals. Way back, it was one of the finest forms fill seal technology they brought in the IV bags. And two products were found to have fungus in the market, one in Shimla and one in Jaipur. And the case went to NHRC because they have found that infusion bags, plastic bags, form fill technology, which was an imported plant, no reason for leakage, that there is a pinhole. There was no reason, possibility in the product. But two bags were found, one in Shimla and one in Jaipur, due to which the case went to National Human Rights Commission, where I was asked to report on the stability issue for these form fill seal products. Dr. Nitin was the chairman of that committee. And what it was found that primarily these two bags had fungus because of the pinprick due to paw of the rodents. And rodents were found everywhere, in the chemist shop, wholesaler, even the manufacturer's warehouses. So if there is a pinprick due to any rodent paw, nail, it can lead to a hole from where the air can enter and lead to fungus growth. So these type of issues which can influence the stability of the product when it reaches and remains with whether it is lying in the hospital store, retailer, wholesaler, or during the transport, and with the patients, all where possibility of mishandling is there. Still, the onus is on the manufacturer to make sure the product is stable till it reaches a consumer. I give you some examples of exports when they happen. You can see over here, these are all pharmaceutical product packs, all fully exposed to sun. Although they are packaged nicely before they are loaded into the, let us say, DHL or whatever the cargo plane. But you will see that they all, I don't know how long they have been lying in the sun. I have seen at Chandigarh airport blue drums of amoxicillin API lying for 1 or 2, 3, 4 days, I don't know how long, in complete sun before they were loaded into the aircraft. So this is very routinely happening, that your whole environment can influence your product. And deep summer, if the products are exposed to sun, the temperature inside can go high. Now, this is, you can actually identify this mountain. This is when I was in the Dr. Vivekanand taking me to from Nasik to Chandwad for one of the SNJBs. And you can see that I was sitting next to him and I immediately pulled out my mobile and the truck was ahead of us. And this had medicines inside and it was a whole solid body truck. And you can see the shadow, how much of the sun is there. And believe me, people have done the study that in the solid body truck, when it is going from, let us say, Nasik to any other city, which is, let us say, 1,000 kilometers away, it is being maybe sent to Punjab or somewhere in the north to Delhi or Agra or wherever, and like from here to Dhule and then to north side. If it is there in the whole sun for 8 hours a day, because these truckers will take rest in the night and they will move in the morning and whole day it will be exposed to sun. And people have found, there was a study done by Panacea Biotech, that the temperature inside such solid body trucks can go as high as 70 degree. Your product is supposed to be kept below 30 degrees centigrade. And this transportation can lead to the temperature rising because this is a solid body truck. And the temperature inside it can be stored because it's a closed from all sides. There is no dissipation out of the temperature which gets inside. The heat gets inside. So the temperature will sustain to 70 degree till it is on the road in the sun. So even intercity distribution steel body minivans is happening. This is, I clicked the photo on Bombay Street, Mumbai, one of my visits to Mumbai. Even Chandigarh, the same, I have seen multiple of trucks, minivans like this. Now, very interesting was another picture. I was in a car and this was going to Naipur, and this was there is a bridge outside Naipur, and I saw this sick gentleman who is taking drugs from the wholesaler and distributing to the retailer. You can see how he is carrying on his thighs all these pharmaceutical products, which is totally illegal to do. So he's carrying all these products on the motorcycle for distribution. And again, it is the full sun. Transporting vaccines on camelbacks in Rajasthan. Although they are into these boxes, but still you can understand the temperature outside must be 40 plus that they have to carry forcefully because vaccination program throughout the desertous area has to be done to kids over there. A view of the typical wholesale market. You can see without shirt, the person is standing and handling the products because it is going to be summer. This person also taken out and put on his shoulders. And because it must be very hot, non-AC environment, same as you can see how the products are being handled. Pharma products are handled in our wholesale markets. And our retail shops which are facing east get all the sun. This I clicked around 12 o'clock when the sun already has gone little, but it is a morning sun. It will be, if the shop opens at 8 o'clock, the whole of the sunlight will go inside, and the products which are lying on the shelf, outside shelves and cabinets can be exposed, glass cabinets. And very interesting is this Cami shop, which I clicked from where I used to buy medicines, and I was always interested. There's a rejected 165-liter fridge from home, which has been brought to the chemist shop. And there's a person has put a cooler because in summer in Chandigarh, Mohali, we have a temperature going up to 40 degrees centigrade. So he puts this cooler for his comfort, but discomfort of products because it really spills out moist air, and many products have to be protected from moisture. So here the products have to manufacture some products which are supposed to be meant. And in India, the problem is that our blisters will be lying on the open shelf without any secondary package, and they are exposed to light. So this is our household mishandling. Your drug box, whether it is covered or transparent, God knows, but there is including long duration retention is there at households, and your drug box can be of any kind. So what influence can be there on products? It can be invisible and visible, loss of active ingredient, alteration in bioavailability, loss of content uniformity, decline of microbiological status, loss of pharmaceutical elegance and patient acceptability, formation of toxic degradation products, loss of package integrity, reduction of label quality, modification of any factor of functional relevance like dissolution, release, etc. So there are some are visible, invisible, some are visible. So I'll show you the products which we had been researching upon. This is anti-TB4 drug combination. Outside it will not be visible. So the whole of the red powder rifampicin is spreaded inside the pocket coming out of the tablet. Here again the same 4-drug combination. You can see that the product is coated and it has really busted out from the bottom, because in both cases in these two products we had ethambutol hydrochloride, which is very, very hygroscopic, and being hydrochloride salt, it reacts with the aluminum foil, and you can see it. And when we checked out the foil and put under the scanning electron microscope, there were pinholes. And ethambutol hydrochloride is corrosive to aluminum foil. It creates pinholes, and the moisture comes in, dissolves rifampicin, and it gets spreaded all over, including ethambutol hydrochloride also, which may be crystallized out. And this bursting is also due to the ethambutol hydrochloride, and it takes up the moisture, creating pinholes. And then due to the moisture, the whole of the tablet is bursting. It's not capping of the tablet. Now you see over here, there is a puffing of blisters. This is amoxiclavulanic acid, which is very moisture sensitive. If there is a moisture left in the tablet, if LOD is not controlled, loss on drying limit is not controlled, what will happen is that carbon dioxide is released in the pockets, and there is a puffing of the pockets or the bloating of the pockets. And this was, incidentally, I was very careful as pharmacist when I went to hospital and looked into the drug tray when she had undergone the surgery at the hospital due to fracture of her thigh bone. And it was a major operation, and her drug tray had this formulation made available from the hospital, and this had such a puffing. It means the drug has decomposed, released carbon dioxide, and the beta lactams can do that because carboxyl groups are there, and this is drug-drug interaction or drug-excipient interaction of the product. So, friends, you have to be very careful as pharmacists on the visible or invisible nature of the changes that can happen on instability. So, you have to be given a good training by your teachers on what wrong can happen to the products. Question is, stability indicating methods will come later to assess what is the content loss. But these physical changes, as a pharmacist, you should immediately be able to check. So we have to understand here what are the factors that negatively influence the stability related quality attributes and stability results. First are the environmental factors like high temperature, which I told you about. If it is a solid truck, same will happen to solid body train if the products are being sent by train. I go to Chandigarh railway station. There is a huge manufacturing happening at Baddi near to Chandigarh, and lot of pharmaceutical products are always shipped by train throughout the country, length and breadth of the country. So there also the issue of temperature, and you can go into the zone where the it is dry in our area, but it is raining somewhere else in the country. So both temperature and humidity is going to be affected. If packaging is not sound, oxygen can get into the product and cause oxidation reaction and then decomposition of the drug. And if it's sitting in a blister, which is not opaque, that's why we have alu blister today, but earlier PVC blisters were transparent to light, and there was all the possibility of color fading. Even bottles get color faded if there is a syrup or paracetamol syrup or like Gelusil, it can lead to fading of the color. And these are all environmental factors. Then we have lot of processing factors which are responsible for instability of products. Poor control during processing of drug substance or drug product, for example, size reduction, mixing, drying, compression, containerization, etc. And these, if they are not controlled, like moisture is not controlled during the whole processing, the product, if it is a light sensitive, in fact, if it is very, very light sensitive. So you need to have sodium vapor lamps. Otherwise, any other lamp will cause decay under the light. So whole production area had to have sodium vapor lamps. So then residual inorganic impurities can lead to catalytic reactions, which are coming from the machines. Then significant amounts of water or residual solvents, if they remain in the product. Significant changes in particle size, distribution of surface area, because surface area can lead to increased surface area can lead to more of the capture of the moisture or exposed to light or even oxygen. Then there can be change in pH, which you may not be aware that many of the products which are liquid products show change in pH. Ophthalmic products show change in pH on storage. Exposure to light, oxygen, etc. Microbial contamination can also lead to microbial growth within the product, and you will never know if a product like Jalusil, which is very particulate suspension, you will not come to know whether it has microbial contamination because it contains syrupy sugar also. Also including a scale-up changes of any kind in this whole processing factors, because you have set the product stability with respect to the pilot scale batches. You have not gone to the commercial batches, and if you go to the commercial batches, you are not gone to the full scale stability testing. So if you make any changes at the scale up, so the stability expiry date you are set at pilot scale studies may not be holding at the on scale up. Then we have a lot of formulation factors like drug-drug interaction, if it's a fixed dose combination, drug excipient interaction, drug packing interactions. Impurities in excipients can kill the product. Co-solvents, light lactic constant effect and their impurities. Added salts, ionic strength effect, pH, microenvironmental pH changes, residual moisture or solvents. Now, very interesting are drug packing interactions, which repeatedly are highlighted, that the label you put on the packaged product, let us say you have semi-permeable plastic containers, and the gum which you are using for fixing the label, pasting the label on the semi-permeable containers can transgress into the product and cause degradation of the product or show as a contaminant on the HPLC and stability indicating assay methods. So there are several unknown peaks that can emerge, and you are not very clear how my formulation over the storage during stability testing behaves, because the glue is getting into the product, liquid product, and showing some peaks, unknown peaks. So any unknown peak beyond 0.1% is considered to be not tolerated, and there can be recall of the product. So lot of issues are there over here, and you should be careful about these. And also our packaging material impurities, leachables from glass, plastics, and rubbers. A lot of emphasis is going to be given now on leachables and extractables, and even their ICH new guideline has come on leachables and extractables. I think ICH Q3D. Then drug migration into plastics and rubbers. It's a reverse migration of drug into plastic and rubbers. Catalytic effect of metallics, unsuitable packaging type, and improper headspace. So you want to protect the product inside, and you fill in, let us say, nitrogen, but nobody knows that nitrogen is replaced by air back. If there is any small chances of leak, it is only 10 seconds that nitrogen is replaced by air. Again, the oxygen will come. So you want to protect your product from oxygen, you bubble nitrogen, but suddenly, due to any of the small tightness is less or any reason of leakage, the nitrogen within 10 seconds will get out and get replaced by air. So whole your protection is gone. So these are the formulation factors. And then we have storage factors, improper storage condition during stability testing, errors in handling and analysis of stability test samples, and bad result interpretation. Storage under unacceptable condition means sending anywhere down the chain during distribution, excursion in environmental condition during shipping, which is a very common thing to happen. That's why you need a temperature and humidity control chambers where you put the product during the shipping so that your humidity and temperature remains controlled. And these days, a lot of emphasis is being given to the supply chain management. And this is one area where a lot of students are needed from pharma also in supply chain management, so that IoT, Internet of Things, which keep the when the truck is moving from one place to another, you have a centralized sensor systems in the trucks and a centralized command center where you are monitoring the temperature variations and the humidity variations inside the truck during the transport through Internet of Things. So this is a new emphasis during shipping and control of temperature, humidity, storage factors during the shipping. And like you can see over here, WHO UNICEF study on the stability of drugs during international transport. So in this, there are two examples given: UNICEF to Kampala in Uganda, and from UNICEF in Copenhagen, the product was sent from Copenhagen to Kampala in UNICEF center in 1989, and temperature was exposed, the products were exposed to -2 degree to 42 degrees centigrade. Because Kampala, they sent the product to UNICEF center in Kampala and brought the product back. And they found the absolute maximum temperature went to 40 degree and minimum temperature with the product phase was -2 degree. Same is the case of relative humidity. So the product faced absolute dry conditions of 20% RH to maximum 77% RH when the product was sent as a trial from Copenhagen UNICEF warehouse to Kampala UNICEF warehouse and brought back. So such a variation can happen. And another example is over here of excursion during shipment of a formulation of UK to South Africa. So you can see over here, these were the constraint conditions of 25 to 5 degree, but the product led to -2 again. And there was a excursion more than 25 and also less than 0 degree. Although temperature limits were, let us say, 2 degree to 25 degree to be stored within that time. So these are the realities in shipment from one region to another in the world. And a lot of products from India are moving to 206 countries, including vaccines. And somehow our smaller, medium scale companies are unable to monitor the conditions of shipment from one part of the world to another. So honest clearly on manufacture to make sure that stable products reach the patient post to manufacture, shipment, distribution, and storage. So final onus is on the manufacturers. Now Indian government has brought the marketing people also into the domain of regulations. Earlier they were not, including retailers, wholesalers, that you also have to make your best effort to maintain the required storage conditions that printed on the label. But yet there are no regulations for patients and households that they are also made responsible if the product is bad. Because there is no pickup of samples from the households by government labs for testing. So what is the purpose of stability testing? Essentially is that it provides evidence on how the drug substance and product quality varies with time under environmental conditions during storage and distribution. Hence, it assures that a product maintains intended physical, chemical, and microbiological qualities as well as functionality and aesthetics within its lifespan. It helps to recommend storage conditions, including establishment of shelf life, expiry date, or retest period. Accordingly, it acts as a key to assurance of quality of pharmaceuticals. Now, how do I assure that the patient will receive the product to optimum quality? Now, what is a word key? You assess your home if it is locked through a key. So how do we assure the quality of pharmaceuticals that it will reach the patient which I am manufacturing and through whole of the chain of distribution and storage, shipment, distribution channels in trucks or whatever mode. So only stability testing, proper stability testing program to be followed is the key to assurance of quality of pharmaceuticals. Of course, manufacturing conditions, ideal manufacturing conditions are also there, but non-ideal conditions after the post to manufacturers have also to be made ideal so that the product doesn't lose its core stability specifications. So that was the purpose of stability testing. And what are the objectives of stability testing? Assessment of product content stability, assessment of product container compatibility, comparison of the stability of product made to a modified formula with that of the original product if you are making a change in the formulation, comparison of the stability of product made by a modified method with that of the original product if you change the method, not the formula, but the method of processing, comparison of the stability of product made by new manufacturing equipment with that of the original product. And these are common occurrences in industry. There has to be a modified formula. Marketing team says, no, I want the flavor to be changed, color to be changed because other product with a blue paracetamol packaging is selling more better in the market. So I want your brown blister to be converted to blue blister. So you have changed now the packaging over here. So comparison compatibility of product with a new container with that of the original container, study of the fact of new materials from a new source of supply on the stability of the product. Now there is a very interesting story about this. New materials from a new source of supply on the stability of product. Now what happens, it is not exactly the new example of new source of supply, but the same supply source. Suddenly, one of the senior industry veteran was quoting an example from his company that suddenly the tablets which they started manufacturing, they were manufacturing in routine. And suddenly a batch of formulation was having black particles. They were confused from where the particles are coming. So is it the instability of the product or there is a source of raw material which has these black particles, very small size black particles, which are not easily detected by eye, but very careful look under a lens would show that the product has black particles. So it has found that the raw material, one of the raw materials which they are procuring, has a problem of these black particles. So this he was a manager, senior manager in the plant. He said, I will go myself to the manufacturer of the raw material to find out why his raw material suddenly has black particles. Now the question comes over here is that when he went over there, he looked into the whole process of manufacture, their previous records of batch manufacturing records, everything, because they were taking this raw material in huge quantities from that vendor. So he went to the plant of the vendor and checked out. So eventually what was found, what a critical review of his manufacturing, that within the 100 meters, which is prohibited otherwise, 100 meters, there's a brick kiln which has got established over there within 100 meters of the pharmaceutical plant. Now that is emitting soot, and that soot is being attracted by his air handling units. And because the particles are less than 0.22 micron, they were getting into the air inside the manufacturing area and getting into the product. So this is a criticality for during the manufacturing, that it is not your problem, but problem of the brick kiln that another person has or the team has put over there within 100 meter radius of the pharma manufacturing plant. So ideally, why pharmaceutical manufacturing plants are away from the urban area or the city area, and they are 20 kilometers outside the bulk drug plant or formulation means facilities, industrial setup, because there should not be any brick kiln 200 meters near to the manufacturing plant. But if you are not as such alert at your level as a manufacturer, and you don't see what is happening in the surroundings, and such a brick kiln can come, it can really affect the quality of your product and eventually the quality of the product we manufacture, the pharmaceutical manufacturer. So these are all very common issues in industry, which a student should be aware of, why these products are showing problems and they get rejected. Now, a company in Ahmedabad went down to venture there to sell their products in Japan, and which was US FDA approved. But Japanese, what they did, they had a 3D projection system. They put a tablet on the projection system, expanded that tablet by 1 lakh times, and found black particles and fibers over there in the tablet, and they rejected the whole batch. So these kind of situations are being faced by industry today because of the intolerance to even extraneous material. So here we are talking about assessment of the product stability, assessment of product container compatibility, comparison of the stability of product made to a modified formula with that of the original product, comparison of stability of product made by modified method, comparison of stability of product made on new manufacturing equipment with that of the original product, comparison of compatibility of product with a new container with that of the original container, study effect of the new material from new sources of supply on the stability of the product, comparison of stability with innovative drug product or RLD approved drug product, Orange Book. So all these are the objectives of stability testing, that you carry out stability testing whenever such changes are happening. And there are regulatory requirements for this, that you have to give commitment, and any post-production process changes are happening of any kind, even the source of raw material, you need to carry out again stability testing on those new products or modified products, or even the change of the site, change of the machines. So there are post-approval changes which are listed, and there are guidelines for post-approval changes where you have to carry out stability testing again. Now, interesting part is whenever the new drug is being discovered and new drug product, so the stability testing lasts from, or it is initiated from the discovery to launch of new drug and product. So stability testing is not only on the final product or the API. At every stage of the discovery to development, the stability testing has to be taken or carried out. But the nature of stability testing, of course, will be different. Now, here is the list from discovery to launch and thereafter. When it is a discovery phase, the emphasis is on candidate optimization confirmation, and we do in silico assessment of degradation chemistry of that API. Now see, try to understand. In second stage, early stage development, what we do here, it is all in silico assessment of degradation chemistry, whether the drug is stable or unstable. So here in early stage development till IND filing, we do intrinsic drug stability, solid solution stability, drug degradation pathway, stability reaction or tox formulation. What is intrinsic drug stability? Is our drug rock stable or as unstable as aspirin, which put in water within a few minutes, it will fall by 50%. So you have to, from the very early, if the drug is proven to be stable, largely stable, you don't need to worry during processing. But if a drug is very unstable to light, like nifedipine, or very unstable to moisture, like beta-lactams or otherwise, so you have to take all the precautions through the journey of drug development. So that is where stability testing in real sense, in some form, starts from when the cradle to grave of the product. So late stage development, clinical till NDA filing, drug excipient interaction studies, stability testing first in human, and formulations for subsequent clinical phases, stability testing of scale up and registration batches, in use stability testing. Then we have patent exclusivity phase, when CGMP formulation stability testing, follow-up stability testing. Then when product becomes an ANDA or it becomes generic, comparative stability testing with innovative product, generic formulation stability testing, stability rework on post-approval changes, market surveillance for stability of market product. So whether my product remains stable in the market or not. So I have to pick up the samples from the market and carry out assessment whether my product is stable or not. Now here, this is from discovery to generic phase. And again, it is from early stage prediction here. And then we do actual some solution stability, pH oxidative photothermal. Then we have tox formulation development where we have to put it to subjective stability testing. Then tox formulation, again, when it is a early stage development and we scale up, and then tox formulation, which is scaled up. And then we have accelerated long-term stability testing for drug excipient compatibility, again, same things. And CGMP formulation manufacturing. And then when we go to generic, and then again, we have to do all those studies like stat stability and drug excipient compatibility to prove product remains stable in the market during the generic phase when it comes to the hands of the generic manufacturer. Now, quickly going through the global stability testing requirements. We have multiple guidelines issues which explain the concept, procedures, and protocols that have been developed and issued by international, regional, and national regulatory agencies to help the manufacturers in the generation of valid and acceptable stability data. So, because stability is key to quality of the pharmaceuticals, that's why the basic international regulatory agencies, regional regulatory agencies, national regulatory agencies have come out with the guidelines so that valid stability data is produced. Estimates are produced. So first is the ICH guidelines. Now, it was the first topic selected for harmonization when it was realized that variation in storage condition requirements in different regions was a main bottleneck in export and import of pharmaceuticals between the ICH regions of US, Europe, and Japan. So all other quality topics followed subsequently. So first was the stability testing guideline, which was taken up for, I think the light has gone out. I have attached my, am I audible? Hello?
Yes, sir.
But my light,
Yes, sir.
Your slides are visible, sir.
my slides are visible. Okay, just hold on. I think there's
Is it also?
a light electricity failure that has
Yeah.
happened. Just a minute.
I may appeal to the audience that Guruji is facing some technical problems. Please hold patience for a few seconds till he reconnects.
My slides are visible.
Yeah, your slides are visible.
Okay.
I do not know whether they are working, whether they are changing.
Now I will try to do change. So now is a table of
Yes.
ICH guidelines. So we had these 1, 2, 3, 4, 5, 6, 7 guidelines on stability testing issued by ICH. ICH Q1A2, ICH Q1F, which was withdrawn later, and then ICH Q5C on biotechnology, stability testing of biotechnology products. The founder ICH stability group leader was who developed Q1A, Q1B, Q1C guidelines was Dr. T.G. Beaumont from GSK UK. And subsequent ICH group leader was Dr. Mr. Nick Turner, who was junior to Dr. Beaumont, who retired and gave the baton to Nick Turner. And he was responsible for Q1A, R2, R1, R2, Q1D, Q1E, and Q1F guidelines. I was fortunate to be with them at one of the stability testing conference in London, where all these ICH group leaders were there, and I was in constant communication with them to understand all the ICH activities that were happening. Now, the very interesting aspect is that there is an update on ICH stability testing guidelines. What they have come out recently is with the one of the ICHQ1 draft guideline they have come out with. In this, what is happening is that they are coming out with a comprehensive guideline. All Q1 to QF and Q5C, they are combining into one single guideline. And that has some new features. Basically, they are streamlining the series of these guidelines. They are combining and promoting harmonized interpretation by addressing potential gaps, including relevant stability strategies and innovations to decrease and remove the ambiguities, addressing additional technical issues, including relevant stability strategy and innovative tools that strengthen the application of risk management, and considering inclusion of new topics such as stability considerations for advanced therapies. So all these new concepts are coming in the draft guideline, which was open for public comments. And this is the front page of this guideline, stability testing of drug substance and drug product. Earlier, all the series of ICH guidelines was stability testing of new drug substances and new drug products. So this, now you see the word new has gone. So this guideline, which is a core collective guideline, will applicable to generics also, and it is going to be applicable world over. So this is endorsed on 11th April, draft version of 2025, and the comments were sought up till from public till August end. Now, if you look into the contents of this guideline, it has a very extensive discussion. Something same wording as the previous guidelines, try to collect all them into one guideline, but some are the new things. So they are giving introduction, development, stability testing under stress test and force conditions, protocol design for formal stability studies, selection of batches, container closure system, testing frequencies, storage conditions, photo stability, which is Q1B is now included into this as one of the chapters. Then stability consideration for processing and holding times for intermediates, which are not there in the original ICH guidelines. WH has included it, and they have now put into this guideline. Then short-term storage conditions, in-use stability, reference materials, novel excipients, and adjuvants, which are the complex generics. New adjuvants are being used in the vaccines and all other biological products. So all these issues are into now consideration. Then data valuation in detail, including the important aspect is the extrapolation. And then we have what we call as the labeling, stability consideration for commitments and product lifecycle management, glossary references, and then annexer one is the stability, reduced stability protocol design. Stability modeling is the new thing which has been added over here, and then stability of advanced therapy medicinal products, ATMPs, which they are calling now. So my issue is that you have to now industry has to start focusing on this comprehensive guideline, which has several new elements than the original ICH documents, and harmonized, more streamlined. This document is there. And if the stability strategy for the future, stability testing strategies for the future has to be now implemented, that should be in accordance with these new guidelines. Still, it is in the comments have been received and the comments are being evaluated, and there may be many changes that come in the final guidance. But some of the interesting aspects, one or two interesting aspects, I will discuss subsequently. Now, there is one that is extensive coverage on stability modeling for extrapolation of results. Now the modeling, there are softwares available now, like one is known as ASAP, which is being used extensively by industry for extrapolation of stability results. The regulatory is convinced now that with the new software that are available, we can go for lean stability program. Not that you need to carry out 1 year, 2 year, 3 year stability testing long term before you launch a product, but you can have some modeling software for stable products at least. You can envisage your long shelf life using a set of studies, which are forced degradation studies, and kind out modeling, apply modeling to assess the expiry date of that product through, let us say, 1 month stability testing or 2 month stability testing. So that kind of stability modeling for extrapolation result is now catching up strongly. You can see over under stability modeling, the next provides additional specific recommendation on statistical tools and model to support the user extrapolation and non-stability modeling approaches. This is an Excel 2 in the stability modeling, and they have extensive discussion on the models for stability testing, extrapolation. There are the coverage on advanced therapy medicine products. Again, you go through, they are talking about diverse category of innovative and complex biological products, which include somatic cell therapy, gene therapies, and tissue engineered products, which are never touched in the Q5C on the biological and biotechnological products. So these kind of products, there are specific requirements on stability testing, so they are including those. Then we have WHO guidelines, lot of them. Multiple the tables of WHO guideline. We have guideline on stability testing of pharmaceutical, which was issued by WHO in 1996 on products containing well-established drug substance in commercial dosage forms. Then 2009, they come out with a stability test of active pharmaceutical ingredients and finished pharmaceutical products. In this guideline, I was the expert from independent expert, not from the government side, and I was party to this 2009. And you see over here, this was modeled exactly on the contents of the ICHQ1A guideline. So I was party to. We had several meetings in Cairo and then in Geneva, and where we finalized this guideline. And word to word, we tried to keep the language also similar so that there was no confusion, but there was some added features in the like in-use stability, variations, ongoing stability studies, which are missing in ICH. Because this was on stability test on active pharmaceutical and finished pharmaceutical products, not new. ICH, all guidelines were new, active substance and new finished pharmaceutical products, but where we had on the generics. So this guideline was essentially for generics, where these type of even the hold-up studies in the latest 2018 guideline, which was further revised, this was further revised. And here we had a long-term stability test condition as identified by WHO member states. Whole of the world, every country was given a storage condition based on discussion with the individual regulatory body. And examples of testing parameters were given and recommended labeling statements. These two were never there in the ICH guidelines. So these were new things which were added in WHO guideline. So I had this finalization of WHO guideline EMRO in Cairo 2008, and then I was party to the expert committee in Geneva 2009, which finalized this guideline. So even WHO now in 2018 guideline, which was a modified form of 2009 guideline on use of modeling and prediction determining drug product stability. So next set 10 of 52nd report of WHO expert committee, it says where appropriate summary of additional knowledge and understanding of stability gained from supporting study, modeling, predictive tools, etc., may be incorporated to support knowledge gained from the primary stability program. So now for faculty, I tell you, whosoever is good at maths and modeling can really be an expert, can come out as an expert for to whom whole of the industry can depend upon for predictive stability testing. Who can understand these all modeling and predictive tool concept. which is now coming strongly in ICH guideline and also already exists in the WHO guideline. So this is a time where our friends from academia should help the industry and help in understanding the models given the ICH guideline and in the WHO suggested modeling. So what best, how Indian industry can take advantage, which is a very opportune time for our friends in academia. So there are regional guidelines from different regional groups like Arab states, ASEAN, EAC, East African Community, SADC, LATAM countries who have come out with their stability testing guidelines in different years. And because they have their own, like Arab states, ASEAN is surrounded by sea. It is highly humid. Arab states are more of the deserter states, and they now are converting deserts into the cities, but they have their own requirements. So, although the international guidelines existed, but regional groups also have come out with this various guidelines. So, EMA is European Medical Agency. It also has come out with the guidelines, multiple of them, and this is both for human products and veterinary products. So I am these tables you can find out from the net, and then we have just a minute. Facing some problem of electricity. So then we have plenty of national guidelines, and you can see over here that we have Australia, Brazil, Canada, China, they have issued their own guidelines. India also have IDMA APA, where I was the chairman. We developed the stability test for existing industrial product. CDSU has guidance for industry. Schedule Y also talks of stability of new drugs. And new drugs and clinical trial rules also talk of stability testing. But there is no formal stability testing guideline issued by our CDSU. Guidance for industry is general, but not specific to stability testing for generic drugs. Somehow we are missed in India, although IDMA APA came out with one of the guidelines for existing sustainable products. Then Panama, South Africa, Korea, they have come out with their guidelines. Then US FDA has its own whole long list of guidelines issued by different organizations within FDA, US FDA, and like lack of expansion date of stability data, requirements of expansion dating and stability testing. They have regulations also and the guidelines also, including drug stability guidelines for veterinary and ANDAs, like for generic drugs also, CDR, US FDA has come out with the guideline. That because US FDA has brought out guidelines for generic drugs, ANDAs, and that's why ICH is now provoked to include a change from new to general, all kind of products, whether new or already generics, and revise the guidelines. Now, further comes your, what I will say is guidelines for various products categories. You have guidelines for cosmetics. You have guidelines, these are various international guidelines for cosmetic products from Australia, guidelines for stability testing, international ISO has issued guidelines, and WISA has given guidelines. Brazil for cosmetic products. There is Cosmetic Toilet and Fragrance Association, which has come out with a guideline. The International Federation of Society of Cosmetic Chemists, they issued in 1992. Then we have guideline on quality of transdermal patches, in vitro diagnostic reagents, in vitro diagnostic reagent. Another guideline is there from ISO. WHO has a guideline on supplementary guideline on manufacture of excipients. The IPAC Excipient Stability Program Guide on, again, there is IPAC America's position for conducting SLA stability and excipients. Guideline on stability testing support of changes or to prescription OTC monograph drugs, something not regulated by NDA or ANDA. Then we have, guidance on homeopathic manufacturing procedures, then medical device registration guideline, then medical gloves and dietary supplements guideline from NSF International. So there are multiple guidelines that have been issued worldwide by either regulatory agencies or associations or ISO, so that there's a full coverage of nature of products that are available in market, including medical devices and others. Then we have a lot of guidelines for storage and distribution of pharmaceuticals. There is a WHO guideline of 2011, model guidelines for the storage and transport of time and temperature sensitive pharmaceutical products, which every pharmaceutical manufacturer must follow worldwide, but Indian manufacturers don't care for it, many of them. Then we have transport route profiling qualification is again a WHO technical report series of 2011 issued in August 2014. And it says in Action 9, model guidance for the storage and transport time and temperature sensitive pharmaceutical products. There is a one from the European Commission on good distribution practice of medicine products for human use, issued on 5th November 2013. Then USP also in chapter 1079 talks of good storage and distribution practices for drug products. Then there are the requirement for stress strategy and determination of intrinsic stability of the drug. That is now part of the core stability testing guideline of ICH Q1 AR2. And both current ICH and WHO guidelines start with mention of stress testing. So you can see over here, first under the guidelines, drug substance and drug product, they are talking of stress testing. WHO says stress testing word both places. In ICH, it is photostability testing. Then the existing guideline at this moment, which is available other than the draft guideline. So the normally stress testing standards, we have been talking about stress studies, stress decomposition study, force degradation studies, force decomposition studies, etc., etc. And regulatory definitions of stress testings have been there for drug substance, drug product. So normally studies are undertaken to illustrate the intrinsic stability of drug substance. Such testing part of development strategy and is normally carried out under more severe conditions than those used for accelerated testing. So on drug product, to assess the effect of severe condition on the drug product, such studies include photostability testing and specific testing, such as, for example, meter dose inhalers, creams, emulsion, refrigerated aqueous liquid products. Now, I have specific chapter today to discuss with you on stress testing and determination of intrinsic stability of drug. This is an area where academia can get involved easily. A lot of publications are coming on stress testing, development of stability indicating assays, and so on. So these are coming from academia and seldom reported from industry because they want to disclose their stability indicating method, which they have reported to US FDA for approval. So this is one of the interesting field for the academia. So it means subjecting drugs and their packaged products to harsher conditions of catalytic external and internal factors such as heat, moisture, pH, oxygen, metals, physics, etc. The samples are collected from time to time and subjected to analysis to identify and quantify the deviation products formed and to characterize the same. So you develop a method, stability indicator method, to identify and quantify the deviation products formed and to characterize the same. So there are coverage of stress testing and analytical methodology in multiple guidelines available worldwide, like FDA guidance for industry, analytical procedure and method validation. So stability test of existing active substance is CPMP guideline. Guidance and stability test of existing drugs from Australia. So they all talk of stress testing and force decomposition studies. They are mentioned in pharmacopoeias, like Indian pharmacopoeias also mention of in general drug products have a test for impurities detected from that in the monograph of the active ingredient in excessive modulation for including degradation products. So identity of such impurities done from stability studies, force degradation study, and analysis of routine production batches. Now, this is the reason that you have to understand why the title of this theme of SNJB 2000, because this type of studies can be done, method development and finding out what are the deviation product, characterize them, are more relevant to academia and teachers in pharmacy colleges. So the trend is aligning of global, regional, and country requirements for stress testing to ICH, WHO, including limits of degradation products. Stress testing being prescribed for different stages of drug discovery and development. Extension to products, including defining stress conditions of specific product categories. Emphasis on mass balance and format for presentation of data. That all is outlined in different guidelines. And there are some suggestions in regulatory guidelines. Nature of stress testing should depend on the individual drug substance and type of drug product involved. Stress testing may be carried out on a single batch of the drug substance because it is an intrinsic type of degradation behavior of a molecule. You don't go for different batches. Should include the effect of temperature in 10 degrees centigrade increments like 50, 60 degrees except above the accelerated testing. Then humidity, for example, 75% of RH or greater will appropriate oxidation photolysis on the drug substance. The testing should also evaluate the susceptibility of the drug substance to hydrolysis across a wide range of pH values when in solution or suspension. Photostability testing should be an integral part of stress testing. It may not be necessary to examine specific certain degradation products if it has been demonstrated they are not formed under accelerated or long-term storage conditions. Results from these studies form an integral part of the information provided to regulatory authorities. So these are essential studies which academia can easily carry out. The detailed information specific instructions for conducting stress testing are almost non-existent in regulatory guideline except WHO TS 929, which really gives a table for typical stress condition in pre-formulation stability studies. So you have to follow these conditions and concentration of API to be used and time duration for stress testing. So you subject stress factor as heat, humidity, acid, base, oxidation, photolysis, and metal ions. So protocol is given in the WHO guideline, but ICH and none of other guidelines give the protocol for stress testing. So it says also WHO guidelines says solid state degradation can also be considered for APIs exposing a solid sample to elevated temperatures such as 60 to 120 degrees centigrade or 5 to 10 degrees centigrade below the melting point can generate a different degradation profile. This approach usually generates degradation profile that can be used as a worst case to assess the performance of the analytical method. So how do you develop an analytical method? By carrying out stress studies, taking it to near to the melting point, pyrolysis, subjecting the drug to near pyrolysis condition, and seeing that your drug is separated from the degradation products. So ICH, which was withdrawn, had a very good feature that 50 degrees centigrade at ambient humidity to cover extremely hot and dry conditions, at 25 degree centigrade, 80% RH to extremely high humidity condition, one batch for 3 months. So these were the conditions they have proposed. Although withdrawal reason was very different, but these are the two kind of studies that can still be followed by the academia. Now, in the new guideline, which is a draft of 2005, April 2005, what they have done is they have differentiated the term stress condition and forced degradation conditions in the latest ICH draft comprehensive guideline. So what they do is stress conducted under stress conditions, they are more severe than the accelerated condition, but not necessarily intended to deliberately degrade the sample. So they have totally changed the whole concept in the new guideline, which is coming up as a draft, which has come as a draft now under comments. So they have added two definitions, stress conditions and forced degradation conditions. Stress conditions are more severe than accelerated, but not necessarily intended to deliberately degrade the sample. So they have added the definition of forced degradation conditions are intended to deliberately degrade the sample, such as elevated temperature, humidity, pH, oxidation, agitation, and light. Earlier, we were taking them as synonyms: stress study, forced degradation study, forced decomposition study. But now they have categorized them separately. The very purpose they give is that development and studies under stress condition is for shipping and excursion outside the condition defined on the labeling. So the understanding of product knowledge and the data gathered from these studies can be useful in addressing unexpected excursion outside the condition defined on the labeling, excursion outside of a labeling claim. So here, what I have indicated to you that post manufacture, when it goes to non-ideal conditions, to evaluate the excursions, which are like 2 degree to 42 degree, minus 2 degree to 42 degree, they have said that stress conditions should be for evaluation of the definition of stress. They are saying for evaluation of what can happen to the product upon excursions. And for forced degradation condition, they are saying the regular stress condition for establishing the degradation chemistry of the compound. Now, this is a very interesting development that what was a synonym, they have separated out the two. So, because that's why I've indicated what are the separate set of guidelines of stability testing and for shipment and distribution guidelines. Based on these, they have now changed. Initially, it was test studies in both drug substance and drug products. There was no forced degradation or test study separate. Now they have separated the two. So academia can do both research into stress conditions to look into the excursion, what can happen. So the increase beyond 10 degrees centigrade increase beyond the long-term accelerating condition will be used for excursion. But if you will take the drug molecule to 10 degrees centigrade less than melting point to establish the stability in the assay, that will be defined now as a forced degradation condition. So this is very interesting that you have to keep in mind that the whole of the thinking or the global agencies is changing to define separately stress studies and the forced degradation studies. So many non-regulatory protocols have emerged because the ICH or WHO guidelines were not giving the details of how to conduct stress testing. In the regulatory guideline, no details were provided except the WHO. So first, we were the first to introduce guidance on conduct of stress test to determine and stability of drugs. Myself and Monica Bakshi sat down to write a guidance document in April 2000 as a 2000. Then there was a rush to publish guidelines and papers on these stress tests. So you can see over a stress test benchmarking study came out in 2020 and 2000 and 202. After our publication, they said how Saranjit has published and we are not doing anything international. People thought of from Bershi, and these all publications came out. Available guidance and breakthroughs we were conducting degradation study. Then Bershi came out with, even my friend Steve Bershi, he came out with a book also, which went into second edition. And then we came out also with in 2013 this publication, Trends in Analytical Chemistry, forced degradation to assess the stability of drugs and products. Now, somewhere we were using for stress studies, stress test definition. Somewhere we are using forced degradation, taking them to be synonyms. Now they have changed the whole concept. That stress studies for excursions and regular forced degradation study for establishment of stability in degrading assay methods. So here we are right. Forced degradation has a safety of drug products, which lead to the development of stability in degrading assays. So this paper, I have always been telling my friends from academia to follow this paper, which was published in Advanced Drug Delivery Reviews by a team from Pfizer, Global Research Development, by Karen El Sante et al., which is a role of degradation for degradant profiling active pharmaceutical drug products, in which they have listed in different development forced degradation timing, pre-phase 1 to 6 to 12 weeks, or after lab experiments are complete, and target is initial IND, formulation development, phase 3 ICS stability start, milestone registration. So this was step 1, step 2, step 3, step 4 of drug development, discovery to development. They have talked about what kind of stress studies are required. And they have given protocols for that. Acid-base stability, when the API is solid or solution or suspension, or drug product is solid, stable at capsule or solution. So what kind of stress conditions should be applied? And then they have recommended API and degradation product degradation conditions. like acid-base hydrolysis. Then for drug product, this is the there. And then we have come out with a recommendation for all our solids that you can use solid acid stressors and the basic stress. In one of our publications, we are given this. And their hygroscopicity, like you can use oxalic acid, fumaric acid, they're all acid stressors. So for solid state studies, like you have drug plus solid state stressors, here they are talking of when it is tablet or otherwise. Drug product when the non-decisive DP is in not solution form. So this is where they say acid-base hydrolysis, or but for solid state studies of acid base, when it is in the solid state, these are the possibilities, buffers, and acidifying agents and the basic stressors. So we have found out that when we do the studies under solid state conditions, sometimes degradation paths are cooperated bisulfite under solid state stress condition. The dotted are for the stipulated pathways. So what we found was that only one product was formed in acid hydrolysis condition in solution. But when we took a solid buffers, and we found more number of degradation products, up to DP 8. Drug product, degradation product 8 were formed instead of 1 in solution. So the solid state degradation chemistry can be totally different. We have this characterization degradation of amorphous and polymorphic forms of cooperative biosynthesis under solid state conditions. So this is our interesting finding, that more degradation product can be formed on solid stress testing than liquid stress testing. So this paper otherwise by Alsante et al. Gives oxidative degradation, what stressors can be used, transition metals, which can be used to stress decompose. Then for drug product or DP in the solid state, pressured oxygen, how it can be, oxidative studies can be done for solids. Then photostability degradation of ICH options to UV visible exposure, that is given. Thermal humidity degradation, temperature 50 to 70 and relative humidity 25 to 75%, and duration also is given. So this is all what Alsante being from industry and Pfizer being the leading innovative industry. So they have published the paper, what we use in stress condition, which we have been using. So following this paper, if you develop a protocol for stress testing or now forced degradation studies, because now in the new ICH guidelines, they have different role of stress testing and forced degradation studies. So you can develop your protocol based on these, and you can carry out studies and report the results in literature. I'm leaving this. There was a Brazilian development of 2015 that they came out that the results must have greater than 10% degradation per condition. It is a possible that drug may not degrade in a specific condition at all. But what Brazil wanted was in 2005 by their RDC 53/2015, the results must have greater than 10% degradation per condition. So there was sudden hue and cry, like from Mr. Myers Squibb. These people said impact of the recent issues and weather resolution on pharmaceutical small molecule forced degradation study requirements. So there was a hue and cry with the leading international agencies that Brazil has imposed something that 10% decomposition in the solid state must have happened, which is not always possible. So Anvisa came out with a protocol of stress condition for drug substance as well as drug products. So eventually, this because the hue and cry was raised by industry, now this resolution by 2017 it was revoked by collegiate board resolution, and or came out in 2017. This 2015 one, when they wanted to make some changes to post registration changes, and the deadlines of this resolution for was already registered. So they revised the applicability of the 2015 guideline, but finally it has been revoked by the collegiate board resolution of 2025. So now in this to February 2025, they say establishes the general requirements of conducting forced degradation studies on medicine containing synthetic and semi-synthetic active pharmaceutical and define the parameters for the notification, identification, and quantification of degradation pertaining these same products. So these all This has established the general requirements of conducting. So they have come out with a new one, and the earlier one have been revoked. So these have been repealed. The following are hereby repealed. Resolution of the Collegiate Board of 2015 and 17 have been repealed. And now they have come out with a new, which is exactly matching the ICH guidelines. So limits of notification and qualification notification in medicine. So sometimes regulatory agencies impose their requirements, but it took 10 years for them from 2015 to 2025 to revoke what decisions they have taken, which was protested by industry. That's why ICHS thought of to include all generics and all other new and drug products and generics into one guideline, so that such occurrences don't happen in future. So still, our suggestion for any future such audit is that resort to comparative studies, drug plus stressor, then placebo excipient mix, placebo plus stressor, placebo plus drug, placebo plus drug plus stressor, placebo stabilizer plus stressor, and then you have a formulation, and then you add the stressor. If you have a complete set of studies, you can justify to regulatory agencies that my drug is not degrading to 10% in some of the situations. And here I say temperature, humidity, acid, base, oxygen, light, metal, and etc. Solution and solid state for drug and non-solid formation. Solid state alone for solid dosage form. That's a question mark. But all these studies, if you have a full data as a pre-formulation setup, and if academia can do such kind of studies, so nobody can challenge in literature. So this is a good opportunity. Now I have retired, but you people, young people, have a big opportunity to do systematic studies of stability testing and stress testing. So you see over here now there is a this was paper published in 2022 assessing the relevance of solution-based stress testing of solid dosage from drug products. So if you have a tablet, why you carried out solution-based stress testing? So here they have a cross-industry benchmark study. They from a lot of companies which are innovative companies, they have come out with that whether for solid tablets you need to do solution-based stress testing or not, where it is relevant, but it's not relevant. So they have come out with the drug discovery development interface, cross-industry benchmark study, how industry, innovative industry is doing and handling this situation. So this is worth reading. So one has to keep a tab on what is happening in the world with respect to even forced degradation and stress testing studies. So benefits of stress studies outlined in ICH-Q1A are two. Stress testing of the drugs can help identify the likely degradation product, which can in turn help establish the degradation pathways and intrinsic stability model can validate the stability indicating power of the anti procedure used. So friends, this is where my whole lecture, the crux is that you have to carry out stability testing program. You really have to establish the degradation pathways, develop the stability indicating assay method, and validate the stability indicating power of the anti procedures, which are supposed to be the regulatory requirements. If these are not met, and in our industry, many of the industries are failing on this, due to which recalls are happening and warning letters are being issued. So we academia should help as much as possible. So I'll take a few minutes more to how we could successfully characterize the degradation product and run the program for characterization of degradation product in our labs. So there is a very important concession in WHO 2018 stability testing guideline. For an API, the following approaches may be used under the stress testing. When available, it is acceptable to provide the relevant data published in the scientific literature to support the identified degradation products and pathways. When no data are available, stress testing should be performed. So our approach was that if we publish one study on a complete degradation chemistry, then the generic industry individually don't need to repeat those studies. Because if we have a good relevant data published in scientific literature to support the identified degradation process and pathway, you just take that paper and attach to their dossier. You don't need to carry out the individual studies by every company in India. They cannot afford also. They don't have the tools also to do it. So that was our purpose, essentially to help the Indian industry. So that has been a preferred research program in our laboratories. So we had been using the LCMS, LCNMR, not LCI, we could not, we didn't have. The modern approach was earlier it was the drug and degradation product was small. Degradation product had to be isolated and subjected to spectral analysis. Modern approach was use of hyphenated tool, and we followed. We had our LCMS and LCNMR in lab to follow, for which we had really used extensively a critical review on the use of modern sophisticated hyphenated tools in the characterization of impurities and degradation products, and critical practical aspect in the application of lithium mass spectrometry for the correction of impurities and degradation products. So this we had really done marvelous work by students in our labs and using and came out with the protocols for doing this. And you can see how many publications we have been able to push into the best of the world journals. So using these LCMS and HD exchange studies, even HS, GCMS for volatile and non-volatile degradation products. And then we used to even predict the properties of the toxicity by using ADMET predictor of each degradation product we used to identify using ADMET predictor. And you can see over here, we through basically NMR, LCNMR, online HDS, the use of LCMS systems, LCMS ion trap, TOF systems. We have published a lot of papers in the literature, almost 50, 60 papers we could publish. So LCMS, TOF, LCMS, and online HDX and LCNMS studies on rosy integration in silico different toxicity of degradation products. Companies approach during drug development. Now, believe me, US FDA inspectors have been carrying these papers and going to industry. They find that rosuvastatin is being manufactured over here. Have you seen papers by Saranjit Singh? Are your analytical methods, stability in indicating methods separating the degradation products being reported by Saranjit Singh? Yes or no? So that has been the impact of the work we have did in our lab. So our series of publication, the protocols provided us have catalyzed scientists for taking up this area of research, leading to appearance of increasing number of publications in the literature. Now, today also, I have reviewed a paper. Every paper that is being published on the degradation chemistry of any new drug from anywhere in the world, it comes for review to me. So that has been the impact of the work we have done from our lab. So US FDA auditors have started asking the issue of validating that their stability indicating methods separate the degradation product, even those reported in the literature. That's why they carry our papers and tell the industry to validate. Now, very 2-3 minutes I will take to development of validated stability indicating methods using stress samples. So you have to develop a method which separates all the degradation products. First, you take the individual stress conditions and then create a mixture of these and develop a stability indicating method. So this has been the procedure in our lab. We will stress the drug under different conditions, create a mixture, and see whether there are the same number of peaks originating or additional number of peaks are being generated. Because what can happen, that something which is produced in acid condition, when I mix with alkali sample, these degradation products can further degrade. So, which may not be in realistic situation when we are doing acid decomposition alone. So we have to verify that all the degradation products, not any new degradation product. Although we can characterize also that also can give us a new chemistry, that acid degradation product on alkaline environment can further lead to this degradation product. Further, degradation product of degradation product. So that is a very interesting type of studies that we have reported also in literature. So basically, stability indicating methods are validated quantitative analytic method that can detect the change with time in the chemical, physical, or microbial properties of drug substance and drug product. And that are specific so that content of active ingredient, degradation products, and other components can be accurately measured without interference. So stability indicating method essentially is separating the drug from all degradation products and degradation product among themselves. So that is stability indicating method. So even the ICHQ3BR also talks of stability ending and defines stability ending method similarly. So we published 2002 classical paper of development of validated stability assay methods critical review, which is followed worldwide. And I think there are more than 1,200 citations to this now. And practical steps in development of stability in method. Conduct of stress testing under different conditions is the first step. Development of initial method and application individual degraded solutions. Generating a mixture of degraded samples. Modification of the initial method. Validation of the final method. Applicable to dual method to drug products subjected to accelerated stress condition, and revalidate if necessary, because sometimes excipients can interfere in the peaks of degradation products when you are having product. So you need to revalidate. So these all studies are very easily done in the any of the academic lab where the HPLCs are available. There you don't need, because you're not characterizing the degradation products here using LCMS or otherwise. So this is an example of SIM we developed for ornidazole in our lab. So this is H2O2 peak for oxidative degradation, and all the degradation products were well separated. So select examples in developed in our laboratories. This was on the ornidazole. This is on metronidazole. We published the paper in 2000, as early as 2001, 2003. Then validated specific HPLC method determination prazosin, trazosine, doxazine in the presence of degradation product formed under ICH recommended stress conditions. And this is HPLC, HMS trazone stress degradation period development of validated stability indicating HPLC method. This is on secnidazole. Then this is on ezetimibe, and validated stability method for alendinoate sodium implying ionic hydrophilic interaction chemotherapy coupled with charge aerosol detection. So this detector we had in our lab, so we could do that. So implementation of design of experiments for optimization of force degradation condition, development of stability indicating method for furosemide. And Anvisa Brazil had also come out with the Brazilian requirements of stability indicating methods. This also, for interest, you can study this paper. So, and now finally, the stability indicating method development should lead to quantitative analysis of degradation product, which is a requirement under ICHQ3B in stability samples. That is a final goal, because they want what is the level of degradation products in your products. So here thresholds are there for 0.1%, identification threshold of dependent upon which normally considered as 0.5%, and qualification threshold is again 0.5% going to 0.15% at the minimum. Here it is 0.1%. 0.1% is the threshold for less than 1 gram daily dose, or greater than it is 0.05%. So if it is reporting threshold is 0.05%, we take it. Normally we take it as identification threshold is 0.1%, and qualification threshold is 0.15%. So anything which is above 0.1% has to be identified, or if it is beyond 0.1%, you have to prove that it's non-toxic. That's why we use ADMET predictor and others to prove that it is non-toxic or toxic. So measurement of degradation product level, the methodology and acceptance criteria listed in ICHQ3B R2, and quantification approaches. I will suggest that industry should use biological method validation guideline, which talks of LOQs, LLOQs, because we are again going to the level of metabolites in degradation product analysis. So LC method now forward for trace analysis. This was impurities in pharmaceutical. Recent application of LCMS for the quantification of impurities in drugs. So these kind of publications are, Agilent has come out with a article on this application note. So user specifications for quantitation of degradation products should be finalized by comparison to pharmacopoeial monographs, other existing drug product specification sheets, batch data and manufacturing ability, safety qualification data, negotiation with regulatory agency. Specs may be set tighter than the pharmacopoeial monographs. Specifications for complex dosage form may be set tighter than the simple products. Drugs with pharmacopoeial monographs generally compared with it, without the pharmacopoeial monographs generally compared with the reference listed drug. So level of impurities or degradation products in reference listed drugs or drug products, your level should not be higher than those. So this is how we set the quantitation limits. So last few slides, 1 or 2 slides, the essential of a successful stability testing program in industry, which I'm not touching today. because remaining within the theme of the SJB and Con 2025. But the industrial program is more extensive. This is one that is carried out efficiently, provides valid estimate of shelf life and storage condition covering the manufacturer as well as the consumer risk, wherein correct and precise information data are available and can be presented to the auditor as and when asked for. So this is stability testing program in industry. So one is for academia and another are the stability testing program in industry. So what is the elements of an efficient stability test program? One is the stability department organization. There has to be a team manpower at different levels, resource management because your samples are coming, lot of testing has to be done, lot of methods are to be developed. So you have to follow a robust stability testing designed based on ICH or WHO guidelines. There is need of standard operating procedures, hundreds of them. SOP should be available. Sample management, there are inventory calculation, inventory management, sample chain of custody, who will have a custodian of those samples, because once sample is lost or let us say is mishandled, it is unrecoverable. But the study is lasting for 5 years. So total inventory management has to be done very carefully in industry. Sample storage chambers, storage conditions, time point scheduling and sample pooling management, testing workload management, risk management. Then you have to do sample analysis and take the results, do all the risk management during the sample preparation, right method analysis, stability, mitigating, validating, instrument selection, allocation and management, instrument qualification and calibration, standard injections, analysis of samples, primary metadata integrity based analysis, result archival, result data sheet. This is what USFT and inspectors from auditors look into. Then data analysis, review and approval, stability data valuation, NES, p-value, pooling of data, shelf and trend analysis, identifying source of OS and OT results and their handling, implementation quality metrics, predictive software, and regulatory reporting, FDA compliant reporting. So let me tell you, friends, and finish my presentation with this, that one part is what you can do at the early stage development studies in academia, report the results which are very useful to industry. One is the whole stability testing program to be done in industry. And these are the elements of an efficient stability testing program. So that is all for today. We have written this book, Methods for Stability Testing, edited by Sanjay Bajaj and myself, and a lot of chapters from all the world best people which are available in working in industry or otherwise, and very well sold. This book is sold in and industry has picked up thousands and thousands of copies of this. So all that is all today as a starting lecture for you people, and my best wishes and thanks are done to you all. Thank you very much.
Thank you, sir.
Thank you, Ji.
Few days back, we were wondering that whether we will be missing this year your exuberance and your energy, which you pack in your every year lectures. But sir, I may guarantee that everybody must have felt that complete justice has been done for their patients, and they have learned. They must have listened to every aspect, every slide. They must have watched, and it will take many years to come in terms with these whatever facts you have shown. But definitely, uh, one thing can be said that in your lecture, it has been highlighted that quality isn't restricted to just a formulation, but it extends to the environment from where it comes. In your lecture, you have also highlighted, even though everybody knows rodents cause a lot of menace to food and food products, also the pharmaceuticals, but you have also highlighted the threats which come from manufacturing units, manufacturing equipments and machineries which are used, packaging materials, and then many things which you have addressed in your lecture. It all indicates that as a pharmacist, we have it on our shoulders that we must educate all the sectors, all necessary sectors which come in the work for distributing the medicaments throughout the world. You have also emphasized the importance of checking the stability at every stage in discovery and development, and also have included several guidelines through which we will have to study and understand. And you have repeatedly have mentioned that the academia must be having several roles in helping the industry to overcome the problems of this stability, degradation products, post degradation products, which affects the safety, efficacy, and quality of a product, which should be reaching the end user. We thank you all with our bottom of heart that you have given enlightened once again. You have, as a duty, you have taken it on your shoulder to enlighten our delegates, sir. And the tradition of SNJB PharmaCon continues. It's a request from us that for everybody that they may put on the cameras so that we can have a snapshot with Guruji and make it as a souvenir. It's an appeal to everybody to put yourself on cameras, everybody, every attendee, so that a snapshot can be taken with Dr. Saranjit Singh, sir.
Dr. Santosh Tambe is also here. Every year he tries to attend here.
Welcome, sir.
Dr. C.R. Patil, I could see him.
Yeah, good afternoon, sir. Good afternoon.
He has joined half an hour back.
Okay.
The technical team is requested to take a snapshot and indicate that it has been taken. Okay, sir. Sir, we thank you and pray to the Almighty God for your speedy recovery, sir. And we wish that we may get your presence. We may be lucky to get your presence here at the campus of Chandwad. The purple chairs are definitely missing you, sir, this year.
Sure, I commit that next time, if everything is fine, I will be there.
Thank you, sir.
But
Thank you. Dr. Upasani, sir,
Yes, please.
should Dr. Singh, sir, be allowed to leave?
Yes,
Allowed to take a leave?
yes. Thank you
I'm
very much.
available. If any question is there, I'm still ready to take one or two questions if from the audience. Vivekanand, you are not online. I think you can go to the next lecture now. People are waiting for the next. Thank you. I may be allowed to leave.
Thank you. Thank you.
Thank you, Ji. Thank you.
Thank you.
Sir, we are thankful from the team of SNJB PharmaCon 25, and we wait for the next year so that we may receive you here.
Thank
Now,
you,
there is an announcement from the organizing committee that we will be having a break of 40 minutes, and we will meet after a lunch break for the second lecture. Thank you, sir. Thank you, sir.
Good afternoon, sir. Shall we proceed for second session?
Hello, good afternoon, sir. I am ready. I hope I am audible.
Yeah, yes, sir. Good afternoon. Very good afternoon, sir.
Sir.
We'll wait for a couple of minutes and then
No problem.
we'll proceed with
Welcome, sir.
Yeah, thank you, sir. Thank you.
Welcome on board, sir. You have been very patient in the morning session.
Yeah, I attended it. Very nice session. It was.
Yeah, Dr. Singh has informed me that he will be completing his lecture by 12:30 as he is compromised, but he continued.
Right. No problem.
Hello. Lotus wave.
So let us begin for the second session of this 2 days national level conference SNJB Pharmacon 2025. Respected dignitaries joining us virtually, distinguished speakers, esteemed delegates, faculty members, research scholars, and dear students. Very good afternoon to everyone. On behalf of SNJB Sriman Suresh Dada Jain College of Pharmacy, I warmly welcome you back to the post-lunch technical session of SNJB Pharmacon 25, the 2-day national online conference organized on the theme Practical and Regulatory Considerations for Stability Indicating Methods in Assays. I hope you had a refreshing break and are now ready to engage in another insightful and thought-provoking session. The post-lunch session gives us an opportunity to explore emerging ideas that are shaping the future of pharmaceutical sciences. It's my privilege to introduce our speaker for this session, Dr. C.R. Patil, a distinguished academician and researcher in pharmacology. Dr. Patil is currently serving as professor and head Department of Pharmacology at R.C. Patel Institute of Pharmaceutical Education and Research, Shirpur. With over 26 years of teaching and research experience, Sir has made significant contribution to pharmacological research, innovation, and academic leadership. He has published more than 120 research papers, holds several patents, and has successfully guided postgraduate and doctoral scholars. He is a recipient of National Award for Teachers 2023 from the Ministry of Education, Government of India, and has been recognized among top 2% scientists globally in pharmacology as per the Stanford University survey. Sir has also actively contributed to digital transformation in pharmacy education and research, including the development of MyCalPharm platform for experimental pharmacology teaching and formal training in digital health and artificial intelligence from IISc Bangalore, making him exceptionally suited for today's topic. Dr. C.R. Patil will share his insights on the topic artificial intelligence in pharmacy. Is it helping to transform the drug discovery process? This session will help us in understanding how artificial intelligence is influencing the drug discovery and how pharmacists and researchers can adapt to this rapidly evolving landscape. On behalf of organizing committee, coordinator of SNJB Pharmacon, it is my honor to extend a warm welcome to Dr. C.R. Patil, sir. Sir, the platform is now yours.
Yeah, thanks a lot, Manjan sir. Thanks for this exhaustive introduction. And I'm thankful to CD Upasani sir and my friend Chatpalli sir for giving me this opportunity. Even though we are slightly deviating from the title of the this particular event, I do feel that this artificial intelligence, machine learning, deep learning, all these things are relevant to each of us students. Whatever walk of life you take, you will be having the major component that is coming as a transformation in each walk of the life is artificial intelligence. And that's the reason why it is necessary to know what it is actually. Generally, now what is happening is everyone starts talking about artificial intelligence, but then very few of us really know what basically is meant by artificial intelligence. So I would like to share what is my understanding about artificial intelligence and how it's going to transform the drug discovery and what is going to be the role of all these technologies in recent advances that are happening in drug discovery. Sir, can you confirm whether my screen is visible to you?
Yes, sir. It's clearly
Yes, sir.
visible
Yes, sir.
and you are audible also.
Okay, thanks a lot, sir. Thank you. So, even though just now you heard about my CV or any other person's CV, each one of us is now slightly concerned about whether the way in which we were working on science or say teaching technologies and all, are they going to remain similar in next 5 years or not? Means AI is literally invading all aspects, all dimensions of each field. And that is the reason why we have to be aware what AI is really and how it is contributing to the different processes related to drug discovery. So that is the reason why I thought that what I understood through a 6 months rigorous course of IISc, I should try to share it with you. And then what are my thought processes regarding how it is contributing to drug discovery, that also we are going to discuss. So 1.5 hours is no doubt very short time to understand and go through all aspects of AI MLDL, but at least introductory aspects we will be able to understand, and then we'll be able to justify where actually we should rely on AI, what are shortcomings of AI, where actually we should use the words like artificial intelligence or machine learning or deep learning. Is there any difference between these or not? All these things we are going to see cursorily in this particular discussion. Okay? So let's start with the agenda of this particular discussion, where I will be trying to discuss with you what are basics of AI MLDL. Then how AI MLDL work. This is very interesting aspect. See, AI, the concept of artificial intelligence started long back, almost before 60, 70 years. But then, after 2003 only, everyone has started to talk about AI/ML very seriously. Before that, people were still skeptical whether AI will really work or not. What is the reason for that? That also we are going to discuss here. We'll try to go through some case studies where AI is helping people to discover drugs or carry out some evaluations, carry out screening. So, what kind of progresses are happening in drug discovery, that we are going to try to see through this particular discussion. Then, what are advantages of this? So, this is an open-ended kind of topic. What are advantages of AI? But we'll try to see whether we are really having some advantages in drug discovery or not. No doubt there are limitations also, serious limitations to AI-based drug discovery, and there are concerns also. So we will try to discuss those also by the end of this discussion. And what are future perspectives or prospects for us? Even those things we will try to discuss in short. Okay. See, the most important word here is intelligence. And people since ancient time, they have tried to understand what intelligence is actually. So this very important question, philosophical question, what is intelligence, has been answered by different philosophers in different ways. So before around 2000 years, there was a philosopher called Socrates. And in his days, he was considered to be the wisest man living on the earth. And then he said, why I am intelligent is because I know that I know nothing. So this is a humbleness, a nobleness of Socrates, who said that the knowledge is so vast that it is impossible for human mind to comprehend what it is. And that is the reason, if you are having this understanding that knowledge is unlimited, then you are the most intelligent person. That is the definition given by Socrates. Okay? But in our day-to-day life, we cannot rely on this kind of philosophical definitions. And surely not in these days where computers have started to take over human intelligence. So we'll have to check why actually the thing that started almost 70 years ago, the concept of artificial intelligence or concept of machine intelligence, it started long back, but no one was really using it to that extent to which we have started to use it. So on screen, I'm showing you some pictures of floppy disks. I'm sure very few of the audience, the members of audience, are having age of more than 50 years. Because unless and until you're more than 50 years old, you would not have used this kind of CD or floppy disk. When I was in M-Pharm, we were using this type of floppy disk, which was almost 8 inch by 8 inches size, and it was very delicate. Means if you slightly bend that particular floppy disk, the data in that will be corrupted. That kind of floppy disk we used in the beginning, and its memory was just 1.2 MB. Okay, so in those days, in 1990s, we were feeling 1.2 MB megabyte is a huge data. So that was our understanding about the computer's capacity. In the second semester of MPharm, we started using slightly smaller floppy, which was considered as a revolution in data preservation. And by the second year of MPharm, we were having still smaller floppy, which was around 2.5 inches in size, its dimension. And it was having around 2.44 MB memory. Okay. So for us postgraduates of 1998, 2.44 MB was the biggest data that a computer could handle. Now, if you ask your PG students or even UG students or even a 10th or 12th student, then they're having access to 2 terabyte USB drives. Okay. And beyond this, now in multi-terabyte kind of pen drives are coming in the size of a thumbnail. Okay. Means this kind of data handling capabilities have increased in last 20 years, 25 years. Again, question comes, how the technology improved so rapidly? What was happening? The thing that was happening is people were developing microchips with higher capabilities. They were developing computer systems which could manage to work on a huge data very rapidly. And with each kind of new version of computers, this computational ability was tremendously increased. Same happened about video shooting also. If you consider year 2000, then the highest definition camera, video camera, was only 800 to 600 pixels. Now each one of you can look into your mobile screen. There the simplest mobile is having a camera of more than 2000 pixel resolution. And your mobile that is sitting on your palm is having capacity to handle a huge amount of data that is being developed through video shooting or through multiple apps and so on. Now what has happened is, because of such means we can say technology is growing with leaps and bounds. And earlier, whatever we called as CPU, Central Processing Unit, that is now being replaced by a GPU, Graphics Processing Unit, where our computers have got capacity to handle even graphical data. We have computers now which can handle multiple GB data sets at a time, or terabyte data sets at a time. Technology is still leaping ahead. Today we are having a new kind of processing unit which is called as tensor processing unit. So these tensors are similar to what matrix is there in the engineering. Okay? In mathematics, we learn few things about the way in which matrices are commuted. So based on those matrix commutations, new processing units have come, which are called as tensor processing units. So in last 20-25 years, ability of the computers to handle the data has tremendously increased. And that is the reason why the concept which had arisen in 1970s, and then it has subsided, because in those days a big data handling capacity was not there with the human minds, or computers were unable to handle that kind of data. But as this data handling capacities have increased, today we have started to deal with big data using our own laptops or palm tops or even mobile phones. That is the reason why we are able to talk about those artificial intelligence related models which were proposed long back. Okay? So one thing that you have to understand here is artificial intelligence is not something new. Only the ability to handle data, which the computer technology has given us, that is the reason why we are able to today handle the data properly and take out some patterns out of it, take out some categorization abilities have increased. And that's what is the main power of artificial intelligence. Okay? So even though philosophically we may say that knowledge is very vast and all, but today computers have ability to handle something that we can consider to be a big crunch of human intelligence or human data that can be generated by human beings or understood by human beings. That is the status in which we are living today. And that is the reason why we have to understand what artificial intelligence is and how it is working. Because of these developments in data handling, because of these developments in processing units, people have started to develop computer programs that can play games. Okay? Now, in earlier years, before 2003 or before year 2000, if we can, before year 2023, not 2003, before year 2023, if we consider chess playing games, chess playing programs, all those programs were developed by human beings, where they were drafting the rules by which the program will be playing chess. Okay, are you getting this? Before 2023 or before 2020s, whatever computer apps or games for playing chess were developed, they were developed by programming each line of that program by human beings. So whatever rules, regulations of chess game are there, they were programmed by human beings. But after 2020s, something new has happened. And what was that development? People wrote a program, a computer program, that could understand the game on its own. And then, along with its experience, it went on becoming more and more perfect. Okay? See how the programming has shifted now. Earlier, each command was written by a human being and tested by a human being. But then a new era has come where such a program has been created which will understand the rules of game on its own, and through its experience it will go on improving. Okay? And when such kind of computer programs came into existence, the ability of these chess playing apps tremendously increased. Okay? So whatever names I have shown here, the first thing is Stockfish. This is a computer app, computer program that can play chess with human beings. There is an interface on which whatever moves a human being will be doing with his mouse, the Stockfish program can reply to it with a proper next step taken by it based on the program that has been embedded in it by human beings. That was the feature of Stockfish. But then a company called DeepMind, it developed AlphaZero. Okay? And what was the beauty of this AlphaZero? AlphaZero did not need word-to-word programming by human beings. It was having a program. No doubt it was also a computer program, but that computer program could understand the rules of the game, and as it started playing the game with human beings, with experience, it went on improving its game on its own without human intervention. And that's where artificial intelligence comes into existence. So I think I'm differentiating what is meant by a simple computer program and what is meant by a program that is artificially intelligent. An artificially intelligent program will be having capability to learn on its own. It is having a program code that enables the computer to learn on its own, improve its experience, and make itself a better chess player. Because of this, today it is accepted that human beings cannot defeat the best computer program in chess game. Okay? Or many times people become skeptical if someone starts winning in a live context. These two people are just playing chess with each other, and if suppose one person starts taking very rapid steps in that chess game, then people become skeptical that he is using artificial intelligence related program to play his next moves. Okay? And whether that person is having access to some computer, that's what is checked now. So such skepticism is coming into existence because computers are becoming far more powerful in understanding such rule-based games. The same we can compare with rule-based drug discovery. Okay. This is how normal programming and artificial intelligence differ from each other. Beyond that, as our ability to handle the data has increased tremendously, we are having today terms like augmented reality or virtual reality or immersive reality. What are all these things? These are also some kind of computer programs which are which are using artificial intelligence, and they're trying to respond to human interventions in the real time. The best example is Elvis Presley's one show which is coming up now, or which a few shows have happened now in London and all, where a three-dimensional hologram of this very famous singer, US singer who died in 1970s. His live concerts are being conducted today. And the 3D hologram program, computer program, which creates the 3D hologram of Elvis Presley on stage. And how is that hologram? That hologram can sing in the same way, in the same style as this singer who died in 1970s. Not only that, he can respond to the requests by audience. So technology and reality, appearance of reality, have changed to such an extent today. And this program is going on. Means even if you search on Google a program called as Elvis Evolutions, you will find that such programs of 3D hologram responding to live requests have been created using artificial intelligence and immersive reality platforms now. Whatever we are discussing here, as we are online, you can keep on browsing your mobile and see whether whatever I am telling you is there in the internet or not. Okay, so Elvis Evolutions, if you see the program, you will see what is the beauty of how immersive reality is coming up, and all these things are possible because of artificial intelligence or generative intelligence. Okay, we are going to correlate all these things to drug discovery very soon. Okay, in this talk. Again, a very nice thing happened in 2024. Two subjects, physics and chemistry, and the Nobel laureates of these two subjects, people who got physics Nobel Prize and people who got chemistry related Nobel Prize, all of them were related to artificial intelligence in some way, or all of them have contributed to artificial intelligence in some way. The best thing is about John Hopfield. This guy is a professor of physics. Okay? And long back, he created a program which could retain the memory related to some instructions given to it. Okay? And this was the beginning about deep learning. The concept of deep learning started from this kind of computer programs where a computer was taught using a specific type of programming to read images, identify images, classify images. And such associative memory related architecture which was there in the software, it appeared like this. There are multiple layers of programming codes in that program. Each layer is having nodes. Now easily you will make out that these nodes are not physical nodes. These are some weightages that have been given. Weightages that have been given to a data point that is entering into the program. What we are talking about is artificial intelligence, which exists in the computer program. And how that program has been created by John Hopfield, he proposed that we can have multiple layers of decision making in a computer program where there will be nodes, means a particular function, mathematical function, and then there will be edges which are connecting to the nodes in the next layer. Okay, and such kind of program was built by John Hopfield, a physicist, to solve his problems related to physics. But then such kind of program which could recognize the images on its own. After getting a proper training, that program started recognizing the images, and that was how he contributed to the science of artificial intelligence. Okay. Now comes the next Nobel Prize, which was again given in year 2024. If you see the Nobel laureates, all of them are very bright and young faces. Okay. Amongst them, one is a biochemistry professor, and other two, Demis Hassabis and John Jumper. These are from corporate world. They were having their own company called DeepMind, which was then merged with Google. So what is their contribution now? Their contribution came in just last 8 or 10 years, where, again, let's remember about the chess game. What was the name of that game? First game was Alpha Zero, which was created by DeepMind company. And these two guys were there who were involved in development of that chess game. And now we are saying that they got a Nobel Prize in chemistry. Why? Because similar kind of programming they used for deciphering folded protein structures, okay, or for predicting the folded structure of proteins, complex proteins. Now you can understand how people from different sciences, different dimensions of sciences have contributed to the development of artificial intelligence. Someone who has started a startup for creating games like chess game contributed to deciphering of the protein structures or prediction of the protein structures. Okay, and that's where they used artificial intelligence for this kind of predictions. Okay, and this is the reason why now we are connecting artificial intelligence with drug discovery, because protein design, computational protein design, or prediction of the protein structure, all these things have become very, very rapid and very, very easy because of artificial intelligence today. Okay. Let's look at the contributions of these people. We are talking about John Jumper, okay, who is from Google DeepMind, a computer programmer. Where he has contributed? He has contributed to protein structure predictions using AlphaFold. Okay, so whatever programs they have developed, they have given such names: Alpha Zero, AlphaFold, and all. So this particular paper was published in year 2021, where they said that their software program has ability to predict the protein structures very efficiently based on genomic data or based on protein bank data, and this kind of predicted protein structures can be used for discovering the role of still undeciphered proteins in the biochemistry or drug discovery or disease processes. As gene sequence data is available today, it is possible that based on the gene sequence, a protein structure can be predicted. And that's what these people have incorporated in their program. And from there, they have got ability to predict the protein structures. Let's look at this paper now, the paper which was published in 2023, where a new version of AlphaFold has come, AlphaFold 2. And if you see the claims of this particular article, then this article is saying that the program AlphaFold 2 developed by Google DeepMind has capacity to predict structure, three-dimensional structure of almost all the known proteins of the planet. This entire protein universe can be deciphered using AlphaFold. Before this particular discovery, no one had claimed this kind of mammoth scale capabilities of a computer. But with advent of artificial intelligence related models, the computer programs which learn upon themselves, people have started to claim that their software today can predict structure of all existing proteins in the universe, in the protein universe. Okay. Why this is important? Because the total number of proteins that have been deciphered by this AlphaFold 2 in a shortest possible time is more than 200 millions. Okay. More than 200 million protein structures have been predicted by AlphaFold 2. And this is important because before AlphaFold 2, only 2 lakh proteins have been, or their structures have been confirmed or deciphered. Their structures have been predicted through actual wet lab data. It's by using instrumental techniques like NMR, X-ray crystallography, or cryo-electron microscopy. People could, up to that date, up to the advent of AlphaFold 2, they could work only on 2 lakh proteins, or they could predict the structures of 2 lakh proteins with experimental data. But AlphaFold, within a year, predicted more than 200 million proteins. 200 means almost 2000 lakh proteins were predicted by AlphaFold 2. Such tremendous capacity is being given to scientists by artificial intelligence related models. That is the reason why we have to look at artificial intelligence from the perspective of drug discovery also. So when these things were happening at the background, again, another company, another startup, OpenAI. Okay. Sam Altman is the owner of this OpenAI. So in year 2022, Sam Altman went and met Bill Gates. Okay. Because Bill Gates has led the world in the first revolution related to computer. Okay. Now, Sam Altman met Bill Gates, and he said that he has developed artificial intelligence based chatbot, and that chatbot has capacity of passing examinations which are given by human beings. It can reply exactly in the same way as if a human being is sitting in that computer and replying from there. So initially, when Sam Altman met Bill Gates did not believe in the success of this kind of programs. He told Sam Altman to come back after 6 months and come with a program that can pass the 12th grade biology related examination. Because what Bill Gates felt was, if you ask a computer to solve mathematics related question paper, it is possible that it will be able to solve it easily because it is based on the formula and all. But if that computer program developed by Sam Altman passes the exam related to biology, then we can say that the program is really having capacity to understand in the same way as a human being can understand. Okay. So Sam Altman was supposed to meet Bill Gates after 6 months, but within 2 months he came back. He met Bill Gates again, and he said that he is ready with the program that can pass the biology related examinations. And literally, whatever app he had developed, whatever chatbot he had developed, that was made to go through biology related examinations. And to surprise of Bill Gates, that particular program passed the examinations with very good marks. And that's where Bill Gates proposed that the new age of AI has begun now, because the computer program is having capacity to understand even biology related questions, and in a very perfect manner, it is giving us the answers related to biology related aspects. How this is happening now? Is the computer really understanding biology? Or it is doing something, it is arranging a data in a specific manner and it is giving it to us? That's what we have to answer. That is what we have to understand by looking at how artificial intelligence is really working. Okay. So again, let's come to this basic question. What is intelligence? As I said earlier, we cannot rely on the answer that was given by Socrates. It won't help us now. will have to define our intelligence. Otherwise, we will not be able to tell what is artificial intelligence. Okay? And such efforts were made by computer scientists long back in 1990s, where considering the definition of human intelligence, a definition of machine intelligence was drafted. Let's see what is human intelligence actually. When we call someone as intelligent, what we are really trying to say from that particular word. If suppose a person has ability to learn, if he has ability to understand, if he can retain the knowledge, if he can apply the logic, plan for something, think in a critical and creative manner, and solve problems, and most important is if he can adapt to the situations, then such an individual will be considering as an intelligent person. And that's what intelligence is mean for us. In the same tone now, regarding a computer system, if suppose a computer system has ability to act appropriately in uncertain environment, where appropriate action is that which increases the probability of success, and success is the achievement of behavioral sub goals that support the system's goals, then we can call that particular machine to be intelligent machine. It's not very difficult. If suppose in simple words if we want to say, if we put a human being in unknown situation, he can take decisions on his own by understanding, learning, remembering the things. If a computer program starts doing the same thing, we can call it as an intelligent program. Okay? And that's how definition of artificial intelligence was given. If a computer system is performing intellectual tasks like human beings, what intellectual tasks? Understanding the language, which was not done by computers before this. Learning something, perceiving the speech or visuals, and solving problems. If all these things are done by a computer program, we can call it as artificially intelligent program. And see, when this definition was given in 1980s. Okay? Since then, people have been struggling to propose that even machines can become intelligent, which has become a reality today. Okay? But in 1980s, they were not having access to the kind of computational powers that we are having today. And that is the reason why, even though the understand or the knowledge regarding artificial intelligence existed since last 40 years, its actual applications are visible today because of parallel advancement in computational technologies. Now, let's see what is meant by artificial intelligence. Just now we have seen what is machine intelligence. The same we can call as artificial intelligence. But again, artificial intelligence is classified into three different categories. The first category is weak artificial intelligence. What is meant by that? It is a very small program and it has limited capacity to solve a particular type of tasks. So all people who are having or who have purchased cars recently, or a smart TV recently, or a smartphone recently, they are having softwares like Siri and Alexa working in their tools. So what Siri can do is it can understand your command and it can trigger certain functions in your device. Same thing is done by Alexa. Very proudly we can ask Alexa to play a particular song from internet and all. So it has been trained to understand your voice and trigger some electronic circuit to start that particular program. That's what is a limited kind of task ability that weak intelligence is having, weak artificial intelligence is having. You'll be amazed to know that whatever progress has been made in artificial intelligence today, it still is related to weak AI. Okay? We are having machines which are considered to be having the weak AI capacity even today. Then there is a concept of strong AI or artificial general intelligence. Even this is hypothetical at present. today we have not reached up to the level of artificial general intelligence. This dream has not yet been realized, because when this dream is realized, computers will be having abilities to solve problems like human beings. Today we are not having such kind of programs yet. Okay. And then there is still more advanced kind of intelligence which is called as artificial super intelligence. So that super intelligence will be surpassing human intelligence, and it will be able to work better than the brightest minds which are available, brightest brains that are available in human race. Okay? But then this is hypothetical kind of thing. Such kind of machines may not come into existence even after 50 years. And that's what is said by experts regarding artificial intelligence. So whatever we are dealing with at present is weak AI and some beginning of strong AI. Okay? So sometimes people may feel, few of you may feel amazed regarding this kind of claims, but this is the reality. Okay? Even though people are saying that artificial intelligence is replacing human beings and all, still it is a part of weak AI which is performing some focused functions. It is not competing with human intelligence even at present. Okay? So we talked about artificial intelligence up till now. We understood slightly what is meant by artificial intelligence. It is the ability of machine to learn, understand, remember, take decisions, and work in unknown situations. That's what is artificial intelligence. There is a sub part of a small part of artificial intelligence which is called as machine learning. So the umbrella term is artificial intelligence, which involves machine learning, which also involves deep learning. So under umbrella of artificial intelligence, we are having all those programs which are developed in artificial intelligence. There are few programs which are developed using machine learning or machine learning abilities. Okay. Now, what is meant by machine learning now? Machine learning is a science that enables a computer to do the things without human intervention, without explicit instructions. A computer starts working on itself, starts learning on itself. If such kind of empowerment is happening because of some software program, we call it as a machine learning algorithm or machine learning model. Okay. Now, even this appears like a fairy tale, right? A computer, how it will understand on its own? It doesn't understand it on its own. We have to train it using labeled data. Okay. So computer is not understanding everything on its own. That's not even possible. What is meant by machine learning is you are training that particular software program by giving it examples of labeled data. Labeled data means what? If suppose you are having CT scans of say 50,000 people, and in those 50,000 CT scans, you are given label as a normal individual, a person with COVID, a person with some lung disease like emphysema, or some lung disease like lung cancer. Like this, you have labeled those images, and then that data you have fed to the computer program. Now, what computer program is doing is it is trying to look at the parameters or trying to look at the characteristics of each labeled image. And from that, it is learning that if such a feature is there in the image fed to me, it is an image related to normal person. If a peculiar part is affected by something, it will be classified as emphysema patient or a lung cancer patient or a COVID patient. Like that, labeled data is provided, and that computer program goes on sorting the data, understanding the features which are categorizing each image like a specific category of patients. If your computer program is able to do this, you call that entire exercise as machine learning. So once it understands what is the feature of lungs, what are the features which will be related to a specific disease, then it will be able to classify the unknown image based on its learning that has happened through the data that we have given to the computer. Okay? So this is what is called as machine learning. Let's try to understand it little better. Okay. So we'll be considering a computer program to learn something. Learn something means what? Learn something from experience. What kind of experience we are providing to computer? We are providing the computer with patient records. Providing the computer means what? We are having Excel sheets, CSV data, we are having images, labeled images, and so on. So such huge database we will be creating in a systematic manner. That database will be presented to a computer program. Now what that computer program will be doing? It will be calling each of the images or data points from the memory. It will try to look at the characteristics of data, and it will go on remembering the things. It will go on learning that if such features are there, we have to categorize the image like this. Okay? So based on this experience, the computer is performing some task T. Task, what is the task here? To identify whether the CT scan is related to lung cancer or not. This is the task which has been given to the computer. What is experience that computer is having? It is having data sets labeled by us human beings, and they have been systematically presented to that computer program. And its performance, means its accuracy to predict the disease, is progressively increasing as it goes on analyzing the data, and human intervention is again telling this is correct, this is wrong, this is correct, this is wrong, in that way. Okay? So its performance we are trying to check, we are giving it some experience, and we are asking it to find out which image is related to a disease related person. So if such kind of exercise is happening, we call it as a machine learning program. I hope this is not becoming very complex to all of us. Okay? These are simple concepts. Whatever we are teaching to our students, suppose to a child, you are given images of cats, dogs, and elephants. Every time when you show your child a photograph, you tell them whether it is a cat, dog, or elephant. So what your child is doing? It is learning the features of those photographs, and then if you show some unknown image of that animal, the child will be able to tell whether it is cat, dog, or elephant. The same thing computer is doing in the same way as a child is doing now. Okay? Because the child will be looking at the features. What is the color of that particular animal? What is the size of that animal based on its background? Whether it is having some features, anatomical features or not. Similar things a computer will be doing. If it is doing this means what? Again, there is a software program, a model has been embedded in that computer. That model is processing the data and trying to sort out the images like that. If this is happening, we call it as machine learning. Okay? So I hope I am making it clear what is meant by artificial intelligence, what is meant by machine learning. The complexities do not stop here. Okay? When we are saying machine learning, whatever I told you now, that we will have to use labeled data, we'll have to tell the computer what are the features of a particular disease or a normal person, and based on that, computer will learn. So in machine learning, there are various approaches again. If you look at these words, supervised learning, semi-supervised learning, unsupervised learning, and reinforcement learning. These are the ways in which computer programs are written. Let's try to understand what's meant by the supervised learning. Supervised means what? You will be providing labeled data to the computer. Okay? And based on that labeled data, the computer will be able to either classify the given data or regress the given data. Classification means what? I told you, normal patient, lung cancer patient, then COVID patient, or emphysema related patient. This kind of categorical classification the computer will be able to do based on the data that you are providing, supervised data that you are providing, or based on the characteristics of the figures which you are given. Computer will be trying to find out what kind of relationships exist in that data, whether there is a linear regression related equation that fits to the data or not, or there are decision trees, or such kind of various mathematical models, whether they fit on that particular data or not, that computer will try to see. And then based on the next input, computer will be predicting what should be the output. That is supervised learning. There is semi-supervised learning. Again, if you see, function of this is classification. Based on the training that you are given, your machine learning algorithm is classifying the data, or it is clustering the data. Clustering means what? It is trying to look at some features of the data. Features of the data means what again? If suppose there is an image, your face photograph is there, then your photograph is having some dimensions, like there is color, then which parts are situated in which position, what is the distance of nose from lips, how the eyes are separated, what is the color of hair. All these are called as different dimensions. What is the outline of your face? All these are called as dimensions. And based on such dimension related data, the compute means that program will either classify you as a male and female, or classify images as cat, dog, or elephant, or it will cluster it into living material, non-living material. Okay. Such kind of classifications computer will try to do based on the input that you are giving. Okay. Now question comes, we understood that if we are providing labeled information to computer program, it is understanding. Can it be unsupervised? It can be unsupervised also, because here also that program has been made in such a way that it will try to find out specific features from each data point that has been given to it. Now chemistry people will sure remember that you are using a algorithm called as K near normal, KNN algorithm. Okay. What is that KNN algorithm? There, your computer program has been taught to look at the features of the data and distribute that data into different groups based on the features. Okay. If you look at such computer programs, it is using simple logic like the distance between two data points on the screen, or distance between two values on the XYZ scale. Based on that, it can go on classifying or clustering the nearly lying data points in different groups. So that is how KNN algorithm works in machine learning. Okay. Similarly, you can go for decision making. Even these are routinely heard terms, like you are having, say, forest plot, or you are having various ways in which computer will be taking decisions related to certain aspects. So if you are doing anything like this, and if your computer, based on your inputs, is learning something, then that particular aspect of artificial intelligence is called as machine learning. But the things do not stop here. It becomes still more complex when we look at the term deep learning. Okay? Now, many times I tell my students that in 2017, I had gone to IIT Bombay for some work, and there they were having a lab named Deep Learning Lab. Okay? In those days, I was totally unaware about what is this artificial intelligence and all. So after looking at this word deep, I thought that it must be name of some alumnus who has funded that particular department, and in his memory or her memory, probably they were given this name deep learning department or something like that. But after going through that artificial intelligence related course, I came to know that deep learning is a technology actually where a computer program is having multiple layers of decision making. It is not single level formula that is used for learning. It is a multi-layered formula, similar to the physics Nobel laureate John Hopfield. Okay, John Hopfield created that neural network kind of thing. Same technology is used in deep learning, and people got this kind of ideas by looking at human anatomy. Or if you want to understand deep learning properly, then you have to look at your own eyes. How? When we look at any particular image, what happens actually is that particular image is created on our retina by the lens that we are having in our eyes. Now that inverted image is sensed by the optical nerves. Those sensed aspects of that image are carried to different centers in our brain. And there is a hierarchy at which different characteristics of those images are understood by our brain. And ultimately, after passing the data through the entire visual hierarchy, we make a decision that a particular picture that is being shown to us is the picture of apple. This happens within a very short time, because of which we do not feel that this much processing is happening. But in our brain, even anatomically, all these points have been shown now, where visual understanding happens in so many different ways or so many different layers. And then ultimately, we understand what we are looking at. So same kind of program was created for looking at images, for looking at videos, and people developed a program, or people developed a model or machine learning kind of model, which is based on deep learning. So this deep is related to multiple levels of decision making, extracting the features of each of aspects. Just look at this image. That image is coming up again in one next slide. So we'll try to see how these three things differ from each other again. Okay, in next slide it will come. So I hope I'm making some sense here. Based on this kind of programs, if you are teaching a computer how to recognize an image, then it is called as deep learning kind of program. So deep learning also is a part of artificial intelligence. So this is the way in which we can tell in simple terms about artificial intelligence, machine learning, and deep learning. And just now I mentioned artificial intelligence is an umbrella term, and machine learning, deep learning come under that. In artificial intelligence, what you are doing? You are processing the data, you are making decisions, and you are trying to learn from the examples. This is an algorithm which is used to mimic human cognition or human behavior. That's what is artificial intelligence we are talking about. Machine learning is something where machine starts learning from the data which has been given to it. And what is most important feature here? Its understanding, its learning improves experience. If this is happening, it will be called as machine learning. What is deep learning? Deep learning is a part of machine learning where it is using multi-layer complex algorithms. Each layer in that algorithm is related to some feature, some dimension of the data being fed to it. And ultimately, by using that entire hierarchy of algorithms, the computer takes decision about something. Okay, so this is how artificial intelligence, machine learning, deep learning differ from each other, and this is how they are having different roles in doing small tasks, learning from the data, and taking difficult decisions like identifying someone's face. Okay, so all these capabilities have come to computers now because of tremendous computational abilities that have been developed. And that is the reason why there is worldwide race on making this computer chips which are having better capacities than earlier versions. People are running behind creating computers like quantum computers, Param computer, Indian scientists have developed. So all these things are contributing to handling of very huge data and testing our hypothesis so that computer goes on learning about it. Using these capabilities now, we can have different kind of algorithms, different kind of learning models, like artificial neural networks we can create, convoluted neural network, and each of these is having its own advantages, disadvantages, specific applications. Okay. Now question comes, what we really want to tell out of this? Generally, after listening to this around 30 minutes discussion, people may start feeling why we are looking at all these details. It's particularly because using computers, we are trying to solve simple problems. Let's look at these three examples now. Artificial intelligence, there is a program. This was exercise given in that training program. What you're seeing on the screen is different digits written by people, right? Each one of us write 1, 2, 3, 4, 5 in a different way. Even the slopes related to those lines are different, right? Now, American government wanted to create a machine that can identify handwritten figures. And for that, they used artificial intelligence programming. What they did is they used a database named National Institute of Standards and Technology, where they were having handwritten figures by 50,000 individuals. And those handwritten 1 to 9 were labeled using a computer, means were properly labeled like 1, 2, 3, 4, 5, like that. Okay. Now this entire data was fed to a computer program. Computer program made out how each digit appears if it is written by 50,000 different people. And based on that, the artificial intelligence related program generated ability to identify handwritten figures, handwritten digits, which are being input to it now. So such programs have been created. If you are doing something of this narrow application using labeled data, using a computer program, it will be called as artificial intelligence related program. Okay. Now machine learning. What machine learning is, just now I mentioned about CT scans, labeled CT scans. They have been designed, which there are ways in which you are telling the computer to extract these features to make decisions. And then based on that extracted features, you are training the computer to classify something. If this is what you are doing, it will be called as machine learning. And in deep learning, I told you, you are having multiple programs which are extracting different features, minimizing the complexity of features, and taking decision like face recognition. If you are doing this, it will be called as deep learning. Okay. This is how computers have become able to handle data, learn from the data, predict something in new situations. This entire capability comes under the umbrella term of artificial intelligence. Okay. Check for yourself. Are you there, Miss? How much time I am having?
You're up to 3:20. We
Okay.
can continue.
So 20 more minutes have remained. I hope I am trying to make some sense to the audience.
Yes, 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 sir.
Yeah. So let's try to see what we are doing using artificial intelligence. The simplest thing is what you are doing or you have been doing since your high school years. You can prepare a graph of x and y axis, and using that y is equal to mx plus c formula, you can predict what will be the value of y if x is particular value, or what is value of x if y is particular value, which we call as regression. So predicting the prices of housing, or predicting the how our sensex will change across the next month. All these kind of functions, if you are doing, they are called as regression functions, and we can create computer programs for such regression related abilities. Second thing, what you are doing in artificial intelligence is you are classifying the images. You are dividing them into different groups based on their characteristics. If this particular thing is visible to you, you can easily see that red color has been classified or separated from blue color, and two conglomerates of data points have been created. Actually, this is done by using mathematical formulae and the dimensions or the coordinates of each data point. Similarly, here we are seeing some complex data. So this will be having a different polynomial equation to tell which point will lie where. Now here the data is still more complex. It is almost arranged in circulated manner, and it is intermingling with each other. So here it's possible that you will be having a second degree polynomial, a third degree polynomial, or a nth degree polynomial equation, and it will help you in classifying the data. Beyond this, by using artificial intelligence, you are using decision making, or you are providing some computer program that can learn from the situations and take decisions for you. Okay? And then the last thing that has come up is generative AI, where your computer can analyze your text. It can analyze the text available on the entire internet. It can analyze images. It can analyze the matrix like data that you are providing to it. It can analyze videos. It can analyze speech, everything. So such kind of artificial intelligence related programs we call it generative AI. But basically, they are also doing the same things that we saw in the initial aspects of artificial intelligence: regression, classification, decision making on a video image or a video film, or on a image or a text or a data point given to it. So AI is helping us in sorting the data like this. Okay? Using all these details, the traditional kind of drug discovery was, which was there, like synthesizing one molecule, testing it in animals, then looking at synthesis of multiple compounds, analyzing multiple compounds at a time, or understanding the effect on gene expression, or we can say microchips which are come, microarray technologies, or computer predictions. Now I can ask you a question. Whatever is happening in Schrodinger, can we say it is having some part of artificial intelligence or machine learning? It is having it, because there also some man-made formula have been incorporated into the computers. But now when artificial intelligence is coming, that computer will start taking decisions on its own based on the data that has been fed to it. That's how Schrodinger differs from artificial intelligence related machine learning kind of algorithms. Okay? No doubt, in Schrodinger also we are having some functions which are similar, or which are named similar to what AI is doing. But AI is taking decisions on its own. Schrodinger has been written by human beings, computer programmers, to take decisions from the data. That's how things are differing between conventional in silico screening and AI based screening. Okay? AI uses those all models, but beyond that, it develops its own understanding, and based on it starts doing predictions. Okay? So this image tells us how actually the things are changing across the entire process of drug discovery. Earlier, based on literature, we were trying to find out the things. We were having very small scale experimental data and all. But today, because of artificial intelligence, machine learning, deep learning, and computational abilities, we can handle a huge amount of data related to chemical structures, related to functional groups, fragments of the molecules, and their correlation with proposed biological activity. So, this kind of prediction abilities we are having, because of which the task that required 2 to 5 years earlier can be accomplished within 1 year today. Okay. Then designing of the compounds. This has also become very easy because based on computer programming or artificial intelligence related software packages, you can take decision which molecule or which functional group can be really added at which position, how that chemical reaction should be set, what will be the probable yield of it. All these things we can predict. We can optimize the leads. We can perform preclinical predictions like DMPK predictions and all. We can check toxicity related aspects in silico. Even in clinical trial, which required earlier 6 to 10 years, many aspects have been now deputed to or deployed through computer programs, and the success rate has increased tremendously because of artificial intelligence. No doubt, whatever is happening in the artificial intelligence is not succeeding every time. It is having its own failures, but people have started to claim that yes, we can use this kind of computer related learning, deep learning, artificial intelligence programs to hasten the drug discovery process. This is one small example I will tell you. After that, we will have some 4 or 5 more slides and we will stop here. So recently, a paper was published in Nature Biotechnology where the group of scientists claim that within 18 months, just within 1.5 years, they found out a new biological target for a disease using AI models. They predicted the safety and toxicity of that particular molecule, and they brought the molecule into clinical trials within 1.5 years. So this kind of things have started happening now. Okay. So they use different AI-based, no doubt, not only AI-based, they used conventional as well as AI-based drug discovery, where they tried to find out which particular biological target is involved in lung fibrosis in this particular case. Then again, they used different kind of computer programming models and data mining related aspects, decision making related aspects, from which they tried to shortlist the proposed targets. Then using generative chemistry, where again molecule generation, all these things they did in computers, and they came up with one single disease target and drug molecules acting on those targets. All of them were validated using in vitro in vivo assays, and immediately they started with toxicity testing, safety testing, and brought that molecule for clinical trial within 1.5 years. Such success stories have started to come up. Now we'll have to see how actually this particular molecule survives through clinical trials and whether it really reaches the market or not. That will be a different story. But then whatever required 10 to 12 years has become 1.5 years now. People have started claiming this. Okay. There are many such AI-based tools like Drug GPT, which can help us in designing the drugs from scratch. Many such programs related to drug discovery are given freely to people to further develop on that. GitHub is a repository of such kind of programs where you can get details regarding this. Even in clinical trials, just by looking at the data that is available on a particular class of drugs or whatever data has been collected up till now, you can prepare AI-based methods to predict what kind of toxicities may be encountered in a newly entering molecule. Even this is possible today. So whatever aspects of healthcare are there, from target finding to patient care, clinical trial and patient care, everywhere AI is very rapidly coming in. Okay. So let us look at one simple example. During the COVID period, it was difficult to contact patients. Okay. Patient means people are asked not to come in contact with the infected people. In those days, how to diagnose whether someone is having COVID or not without touching that person or without making that person to come out of his home. There, IISc professors had proposed one program, computer program, that could understand whether the person is having COVID or not, just by analyzing his sound clip. Okay? And how the sound clip was generated? They asked the patient to just create a 5-minute clip using his mobile phone and just post that sound clip on WhatsApp, and that sound clip was analyzed at IISc based on the pattern in which person is speaking. They could correlate whether the patient is having COVID or not. Okay? Such kind of out of world technologies are coming up now. Okay? Because image which is appearing in front of you is a graphical presentation of sound wave. Okay? And particularly in whatever you are seeing on the screen now is a computer image created for the word 19th century. Okay? So you can imagine now a computer has a vision to understand this figure. It can analyze the figure, and from that it can make out which particular sound is coming. So such kind of technologies are coming into existence. But then we have to also understand that AI cannot get everything that we can do. Our role is still there, not still there. Our main role is there in all the aspects related to use of AI, because even though AI recognizes some images, it will not be able to tell whether the facial expression of this cat is like sad or pleasant. Okay? It will not be able to tell which of these demons are large in size. For us, we are perceiving that this background image is the demon that is coming from backside, it is having a larger size. This is our perception. But for computer, both these images are having same size. Okay, so such kind of perception related aspects are always going to be there with humans. We are having our own way of looking at the world, and that cannot be completely programmed in any of the computer programs. Now, whatever we are looking at ChatGPT, we feel that it is intelligently talking to us, but that is not the truth. The ChatGPT app is taking up words from internet. It is just arranging it in a particular manner and presenting it to us. It's not understanding actually. What it has understood is which word can come after which word. But as a whole, it is not having any understanding about what is meant by that data. So we have to be aware about such aspects, such shortcomings of this AI. And we can start using all these things like ChatGPT. You can use very nicely. There is Perplexity AI, which you can use for literature searching, for designing your proposal, for designing your patent. Everywhere these things are helping like anything. Notebook LM is one of the great sources now. You use Notebook LM to analyze whatever huge text you are having. You can ask questions to Notebook LM, sort the data, prepare tables, everything within seconds. So even this tool is very good. Gemini AI can tell you about images. It can classify images, tell the features of images and everything. Okay, so AI is the limit today for all the researchers, and we'll have to look at AI with the no doubt great hopes, but also with some skepticism that it can cheat us even now, because it is not understanding what it is telling us. Okay? So this is what I wanted to share with you. Thanks a lot for this opportunity. And I hope I could create some understanding about what is meant by AI, ML, DL basics and how it is being used in drug discovery. Thank you. Thanks a lot.
Thank you very much, sir, for the insightful and engaging presentation. As usual, we will now open the floor for question answer session. I request the participants to kindly ask their question to respected sir, or they can put their queries in the chat box or raise the hands.
My suggestion is simultaneously you put your queries to ChatGPT also. Maybe it will be answering better than you.
Yes, yes.
All right. If you wish, you can share this presentation with all the participants. Whatever aspects they want to check, they can
Yes,
again
yes,
try
yes,
to learn it through some AI-based tool and try to get some basic understanding at least what is happening in AI ML here.
yes, sir, definitely.
Thank you, sir.
Patil sir,
Sir, please.
sir, it was a wonderful session.
Thank you, sir.
What you have highlighted on AI and machine learning. And I think in Pharmacy Council of India in next year they are going to put the new syllabus, and
Right.
then they have some topic on AI and machine
Right.
learning.
True,
So
sir.
here the question is that
Hmm.
nowadays in pharmacology we are having most of the experiment simulation type.
Hmm.
So by using this AI and machine learning, how will we justify this experiment in coming years?
Okay, see, justification of experiment is cannot be done by AI, no doubt. Ultimately, the real world experiments that you are performing are the final words regarding any drug. Okay? You
Okay.
cannot just predict using AI/ML or some simulation that won't help in real life.
Okay.
What is happening is you can take certain decisions very rapidly, which you could not have done in the absence of AI. And that's how you have to use it as a tool to just accelerate your thought process.
Okay.
But ultimately, decision making will be yours. It will be based on your understanding of how the lab experiments are working really. Okay? We cannot rely on these tools for anything like ultimate decisions.
Okay.
And for teaching also, simulations they help us in packing the data in a better presentable manner. But unless and until you perform the experiment, you do not understand it. That
Right.
is a reality. Ultimate gold standard is wet lab experiment. That's where you have to train people. There is
Yeah.
no option for that. But virtual reality can help you in virtually performing the experiment before you go to
Yes,
wet lab.
yes. Yes, correct. Thank you.
Next speaker is waiting. I think we have to clear the floor.
One question, sir. One question, if you allow, sir.
Please, please, sir.
Uh, what will be the teaching aspects of AI? As AI is about to be inducted in pharmacy course, who in your view should be the right person to teach it?
Okay, now AI programming we cannot expect from BPharm students. Even this
That's well.
opinion we had conveyed to PCI that if suppose by the end of fourth year of their course, 80% of students, if they can use some useful applications for predicting something and all, that should be considered enough. We cannot teach AI/ML to pharmacy students, or our core subjects will be neglected in that case. That's
Okay.
what's happening
Okay,
now.
that satisfies.
Yeah, present AI syllabus, they have given AI for 7 semesters, and they have just categorized it for AI for pharmacognosy, AI for pharmacology. That's not the way in which AI can be taught to students. It will be a burden for them. Just how to use some applications, if they understand, that will be more than enough. And whoever is having interest in AI after that, after BPharm course, they can work on that separately. But for all the students, putting this burden of AI/ML is not justifiable, at least at present. And from
And that for
yeah,
aspect, I consider that the student must be well versed with statistics, biostats,
right,
and mathematics.
right. True, sir. Even after working for 27 years, even we are stumbling at some points. Means we cannot understand what is happening really. We expect it from
Correct.
students is wrong.
Okay. That's true for our subject also. That
True, sir.
happens many times in our subject, in our domain. After
True.
working for 25 years, we
Yes.
feel difficulty in understanding our own subjects.
Right.
And so we can't go into AI. Okay. Thank you, sir. Thank
Okay,
you.
thanks a lot. Thanks for
Over to
this.
the coordinator.
Yeah. As we come to the conclusion of this session, I would like to sincerely thank you, Dr. C.R. Patil, sir, for the clear, insightful, and well structured lecture. Sir, in today's session, you effectively introduced us to the fundamental concepts of artificial intelligence, machine learning, deep learning, generative AI, helping us understand how these technologies different, complement each other in pharmaceutical research also. You also provided a thoughtful, provoking comparison between traditional drug discovery and approaches in AI-based drug discovery. clearly highlighting how AI has the potential to reduce time, cost, and complexity while improving efficiency and decision making. On behalf of SNJB Sriman Suresh Dada Jain College of Pharmacy and Organizing Committee of SNJB Pharmacon 25, I extend our heartfelt gratitude to you, sir,
Thank
for
you, sir.
sharing your valuable knowledge and experience with us. I would also like to thank you all the participants for their active presence and engagement. With this, we formally conclude this session of SNJB Pharmacon and request all the participants to kindly remain connected for the next session of this 2 days national conference.
Yeah.
Thank you. Thank you, one and all. We'll have a short break of 10 minutes and then we'll continue with the next session. Thank you, one and all.
Thank you, sir. Thank you.
Oh, good afternoon.
Good afternoon, sir. Good afternoon.
Sir, good afternoon.
Good afternoon.
How
Good
are you?
Fine, fine.
Yes, sir. Good afternoon.
Good afternoon, Patil sir.
Yeah.
Good afternoon, CR.
गुड आफ्टरनून सर. गुड आफ्टरनून. ठीक होतं काय सर? म्हणजे उपास्य सरांनी काय सांगितलं नाही, कसं वाटलं लेक्चर?
अरे तुझं लेक्चर म्हणजे आमच्यासाठी फिस्ट असते. Because so many new things we came to know.
Very well studied, deep
Ah.
into basics. Basically, a lot of basics are there, which are really good to know.
But this is my experience from so many years. I think if I'm not wrong, I'm learning from CR many a things, and this must be his 28th or 23rd lecture I'm attending.
Okay. Oh, good.
Every presentation, every lecture, he incorporates new things and he keeps on experimenting and what you can say, extending his capacities to deliver.
Thank you, sir. Thank you. Thank you.
There was one lecture, I remember Dr. Bodhankar sir many a times. Myself and CR were delivering lecture in that faculty development program at Pune College of Pharmacy. So my lecture was altogether a boring type of lecture, textual and all.
तुमचं ते याच्यावरचं होतं सर, सेन्सर्सवरचं होतं म्हणजे.
But once CR started delivering lecture, everybody was laughing, enjoying, and Bodhankar sir was so delighted to listen to it. At that time, he told me, या मुलात दम आहे बरं का म्हणे, हा काहीतरी नवीन एक्सपेरिमेंट छान करतो. So this was his compliment.
Thank you, sir.
Good. Meanwhile, I will just share. I have one video to show. Just we would like to test it. Anyway, we have some time. I'll just how to do. I have to share my screen first, right?
Technical team should allow Dr. Prasad to share his screen.
सर बॉटमला जे ग्रीन एरो आहे वरती, त्याला क्लिक केला तर होऊ शकतं.
केला, आता शेअर दिसतंय मला स्क्रीन, एंटायर स्क्रीन म्हणू का?
हा, एंटायर स्क्रीन करायला पाहिजे.
Okay, share. Chala. So now you should be able to see my screen, right?
Yes.
See, something is there, ChatGPT.
Okay.
I had to close this first. एक मिनिट, काय झालं इथे हॅलो आलं ना. So I'll share my presentation first. Can you see my presentation now?
Yes.
Visible, sir.
Okay, now there is one video. एक मिनिट, presentation
प्रेझेंटेशन मोडला जायला. हा.
Can you hear my sound? Just tell me the sound. Video is clear.
Is an ability to sustain physical activity or mental
You
efforts
can hear it,
for a
sir.
longer period of
Can you see?
Yes, yes.
Yes.
You can hear the sound, right?
Yeah.
Yes.
Okay, done. So the point is done. Testing is complete. Okay. So
And,
now we can talk.
and secondly, Dr. CR is involved with artificial learning and artificial intelligence from so many years. He is
Yeah.
preparing those simulated experiments and CDs and videos.
Right, sir.
So he will be always 4 5 steps ahead of us in machine learning and artificial intelligence.
नाही तसं काही नाही सर आपल्याला भरपूर म्हणजे वेगळंच फील्ड आहे ते सगळं काही लवकर लक्षात येत नाही काही गोष्टी यातल्या.
We also need to learn and implement. This is going to be important. We may not be able to do this for drug discovery per se, but in our regular life, there are a lot of
Right, we can use it.
applications.
Yes, yes.
But what I feel is this artificial intelligence is making so many intellectuals in a real sense, they are making all the intellectuals artificial.
But there are some issues also. People are discussing those
They will
issues.
not come forward with their natural instinct. They will come with artificial instinct.
How to unshare the screen now?
Right, sir. How to, yeah, just you have to click on the share screen once again. It
Once
will be
again, I shared it. So content only as background. I don't know how to unshare.
So option may be there, sir. Stop sharing live.
नाही काहीच नाही ऑप्शन इथे. अच्छा माय स्क्रीन तिकडे असेल. ओके स्टॉप शेअर समथिंग इज देअर. येस गॉट इट.
Good afternoon, sir.
Good afternoon.
How are you, sir?
Fine, fine.
Looking great.
Vivek will be remembering that in our PhD, drug discovery was on the peak. This computer-aided drug design. So we use a lot of techniques,
Speak now.
which
It is just a tool.
it was just tool, but I went lot of details.
Instead of synthesizing hundreds of compounds in laboratory,
Yes.
and then testing them in animals, better to predict ahead.
Actually, we could save a lot of animal experiment because there are high throughput screening technologies were developed that time.
Yeah.
And we could screen a lot of compounds within a short period of time because of that high throughput screening. But of course, there was a part of the computer, not artificial intelligence. But I remember in my thesis itself, I use pattern recognition, which is the basics of part of the basic, of course, very small part of machine learning, pattern recognition techniques. So because of even neural network, I wanted to use neural network technologies in my research, but I couldn't figure out how to do that. The awareness and resources were less. So internet was not very developed. So neural network I knew about from that time, and neural network is a base for machine learning and artificial.
Yes.
So these techniques we should use. A lot of good techniques are there.
Yes.
Like one of them, notebook LLM, we already mentioned.
Yes.
There are many such techniques actually. And tools are also coming, but many times tools are expensive. So we have to judiciously evaluate. And whether it is how much is really. Just I wanted to ask you because you are in this area. Uh, is there any good tool to make good presentations?
There are many, sir. Many free tools are available, but they will be creating few slides for a few presentations. But after that, nothing. Even Gamma AI is one tool. Gamma
Okay,
AI
okay.
start working.
Gamma, I know. So what is your opinion? Means I use it. Um, I am not very clear. Means I have to evaluate more in more detail actually.
It depends on what kind of prompts you are giving. If suppose you, what you can do is in Chat GPT, you give your opinion about that topic, ask it to create slides based on that. You copy those contents, then put them in Gamma AI and ask it to prepare the presentation. So it will not be completely AI based presentation.
Mm-hmm.
Your own presentation just polished using AI tools. That way it comes better. Your designs are very good, so it saves much of your time. That's that.
Is some? I think it is paid tool, right?
Yeah, yes. But you use 2 3 packages, but start with your own content. If you want to tell through that presentation, you prepare at least say
No,
one
no,
page.
see, the one thing about these tools, I can tell others also that definitely that one line prompt is not going to work. It
Right.
is sure. I am very sure about it.
Yes.
And you need to have your own intelligence to use artificial intelligence. This is
Yes.
basically the thing.
Yeah,
So
one has to
how
keep on working over it
Yes.
repeatedly.
there are a lot
And
of learning you have to do with machine
yeah,
learning. When
in a
you
quest, what
interact, you
happens
need
is
to
we are,
learn and then improve on
yeah,
your prompt and other areas.
yeah.
Then this will work
And it
well.
happens such that one tends to drift away from their basic research. A single person cannot handle
That
everything.
maybe no, no. That maybe it will be useful sometime. Say this is a like how you use it. That is
That's
important.
yeah. Yes. See,
That will
earlier,
Years
need
back, it
that
was
will
just a
need
mechanism.
really. I'll tell you that will need a experience.
Right.
Many times I learn. Actually, I attended some workshop also on AI on different aspects. Most of the common thing which people told is if you use it irresponsibly, you are going to harm yourself. So people are there who did suicide based on AI.
Right.
So a lot of controversies are there, but ultimate conclusion can be you have to use properly
Right.
within proper for the work and don't believe blindly. That is what I would like to
On
conclude.
the same line, I will request CR
Huh.
prepare one presentation and lecture how to use AI responsibly.
Yeah, that I can do, sir. Yes.
Understand? So that all the people will able to at least understand if we don't use responsibility, what will happen.
Sure, sir. Sure. I'll prepare one lecture on that.
Because nowadays I am finding many people write their emails through AI and they copy and paste.
Right.
So I could get that name and all these annotations, double inverted as it is.
Right.
See, now more than irresponsible will be having harm to the very large extent. Actually, companies had to pay huge penalties and fines just because somebody blindly uh, g- just blindly thought that this report will be correct and they supplied to their client that report generated by AI and client afterwards found that it is wrong, figures are also wrong, and then they have to pay a huge amount of fees for
Right.
to the client. So this kind of harm is financial harm is also there.
Right. Right.
Naturally, other
Yeah,
personal level,
responsibility.
there are many
Yeah,
harms.
in that aspect, responsibility and judicious action cannot be taught in the classes. It should be inbuilt. It should be understood by the person
Yeah, but
himself
awareness has
involved
to be
already.
there. Everybody is responsible for its own
True, sir.
deeds. But
Yeah,
only thing is, one should at
people.
least this awareness should be spread that
But
if you don't do, you are the sufferer. So
but
it's up to you.
I'll say one thing, only for saving time, if you are not using your brain properly, then one day you will forget to use your intelligence.
True, sir.
Yes.
And this possibility is very high in case of these Gen Z type of people.
True. True, sir.
They will simply take help of AI and they can say that, yes, it is this is as it is. It is given in AI.
Yes, true, sir.
So it will be very difficult.
I think this is a transition period, so everybody is trying to adjust to this new world of AI.
Right, sir.
People will realize, and those who can do best use of this time will definitely will get benefit. And of course, the judicial use is a one very important aspect of that thing.
True, sir.
But what I felt very strongly is due to this a intellectual development or using brain correctly or application of knowledge correctly, this thing will deteriorate very sharply.
Right, that's going to happen. True.
Yeah,
आजही आपण म्हणजे कॅल्क्युलेशन करताना 15+17=32
कॅल्क्युलेटर वापरतो.
असं करून टाकतो आपण.
बरं.
आपल्याला ती सवय आहे, पण नवीन पिढी काय करते, ते कॅल्क्युलेटर काढतात.
कॅल्क्युलेटर.
Or otherwise, God knows they may take help of AI also for these additions.
This will be too much. AI is not required. This calculator is that is at least there.
Therefore, here next to next SNJV Pharmacon, one topic is sure for you.
Sure.
How to use AI responsibly.
In fact, we should keep this as a theme.
Yeah.
Theme would say, I can tell you about in our area how we are using and people are using, of course, in industry. CR can tell exactly for teachers how they can utilize, and student how they can utilize that tool to learn. Like what he already mentioned, some of them in his lecture today, so that he can elaborate there. And even some other person can tell us how to some other aspects of AI, maybe ethical aspect. Maybe we should call some psychologist or humanity person to tell us about AI and responsible use of AI. Maybe legal person to how see, because if you don't use it, may be illegal also sometimes. So we should not say we should not involve in the illegal things like copyright violation, or maybe we have to see those legal aspects are also important now.
Right.
Psychological aspect, legal aspect, and then what we are having is application aspect maybe.
It's needed. We can plan, sir. We can plan for this. Yes.
So, Dr. Chatpallivar, we are having a good theme for next SNJB Pharmacon.
Yes.
Okay, sir.
Just get it approved by our mentor.
Yes, sir.
So he also can highlight so many
He
things.
can.
Not only AI. May I add, sir? May I add? Not only AI, responsibility and judicious activity should also be exercised while using simple laboratory equipments, apparatus, machineries. That's what was evident in the morning session when he was displaying
Then
the
this is actually better.
machineries, different
This is
machineries.
better. AI is one part, but judicial use or responsible ethics. I will say ethics in our area, like ethics in pharmacology, ethics in say drug discovery, ethics in day-to-day life or things, or teaching, even teaching. Even clinical trial, there are a lot of ethical issues that I can definitely tell a lot on that. Or publication, when we publish the thing or book publish or any publication, you need to have ethics. Now we know people who use AI to do. Yesterday I heard that some publisher published 50 books in 1 year from one single author. And it is very reputed publisher. It is not very small publisher. It is an international publisher. They are now facing
May
the
I remind
problem.
you, sir?
Yeah, good
May I
afternoon.
remind you, sir? Yeah, may I remind you, Dr. C.R. Patil was the same person who has highlighted this point in one of his lectures here at SNJP campus
Okay.
some 12 years ago. He has highlighted some papers which were actually maybe we may call it fake or predatory.
With
Hello.
AI, it is going to be more problem. Already it had a problem now. We
Right.
need to,
Good afternoon,
so
sir.
maybe ethical and responsible use of technology. This can be a theme.
Shall
Yes.
I please
I
test
think
previous?
we will stop our discussion here. We are getting late for the session.
Okay.
And that coordinator is eagerly waiting for it.
Yes.
Okay.
Yes.
So let us start our session then.
Okay. Very good afternoon to one and all here. Myself, Dr. Naina Baste, welcomes all for today's third session of Pharmacon 2025. It is a matter of great honor and privilege for us to welcome today's third resource person, a highly accomplished scientist, academician, industry leader, and researcher, Dr. Prasad Thakur Desai. Dr. Prasad Thakur Desai holds a PhD in pharmaceutical science from Nagpur University and has over 34 years of rich and diverse experience spanning academia, pharmaceutical industry, clinical research, toxicology, and regulatory science. His core research interests include clinical and molecular pharmacology, toxicology, health sciences, natural products, complementary and alternative medicines, sports nutrition, exercise physiology, animal welfare, medical informatics, quality of life, and community pharmacy. Currently, Dr. Thakur Desai served as Chief Scientific Officer at Indus Biotech Limited, Pune, where he has been leading advanced research initiatives since 2018. Prior to this, he played a pivotal role as General Manager, Scientific Affairs and Quality Assurance at Indus Biotech Private Limited for nearly a decade. His career also includes significant academic contribution as an assistant professor at Pune College of Pharmacy, Bharati Vidyapeeth University, and as a faculty member at BITS Pilani, along with teaching and research roles at Guru Nanak Technical Institute and Blessing Pharmaceuticals. Dr. Thakur Desai is an internationally trained professional in intellectual property rights, having completed multiple certified courses from the World Intellectual Property Organization, WIPO, Geneva, including patent drafting, patent information search, and intellectual property management. His scholarly contributions are truly remarkable, with 118 peer-reviewed journal publications, 67 conference proceedings, 2 books, 9 book chapters, and 11 magazine articles to his credit. He has successfully guided 4 PhD scholars, 43 M.Pharm students, and 21 B.Pharm students, and has delivered more than 75 invited lectures nationally and internationally. Dr. Thakur Desai's excellence has been recognized through numerous prestigious awards, including Best Paper Award at VU Innovate Pharma Connect 2024, G. Achari Prize at IFSCON, Anvos Award, PC Dandiya Award, and multiple Best Oral and Poster Presentation Award at national and international conferences. With his vast experience, visionary leadership, and impactful contribution to pharmaceutical sciences and health research, Dr. Prasad Thakur Desai is truly an inspiring personality for all of us. Today, he is going to enlighten us with a topic fueling with fenugreek, compressing endurance enhancement with Endugor, which is their nutraceutical product. So it's my privilege to invite Dr. Prasad Thakur Desai to join us virtually and enlighten the audience with his expert insight at Pharmacon 2025. Over to you, sir.
So I'll share my screen now. I hope everybody can see the PowerPoint screen. Can somebody confirm me?
Yes, sir. It's visible.
Visible. Okay. So good afternoon, everyone. So this today's topic, which I wanted to focus on one of our product which we develop from basics called as Enducore. Uh, as just we discussed that one of the area where uh, I'm interested is exercise physiology and uh, sports medicine. So in sport medicine, basically we are talking about the supplementation which sportsman and athlete wanted to take. So in Pharmacon 2022, I discuss about one of the complementary medicine, which was one of our product called as Breathmore, which was from cinnamon bark extract, which is a nasal spray. In 2024, I discussed about one of the functional food called as Phenuflex. So these two products are completely different regulatory areas. So today what I am trying to discuss with you is another area I wanted to open in front of you is a sports nutrition, where a lot of research can be done by us as a pharmacist and pharmacy professionals. Because ultimately what we are trying to do is improving our physiology, enhancement of our physiological expertise, possibilities to excel in the sports. Now, many of us are already doing running or walking at least for to maintain our health. Naturally, it gives a lot of benefits to us, which everybody knows. So I won't go in detail. So athlete want to do this thing to next level. They wanted to go to extremes and they wanted to increase their capacities. In this case, it is a endurance. So I would like to start. What is that endurance basically means? It is ability to sustain physical or mental efforts over a long period. So in very crude language, it is called a stamina. But this is much more than that. Stamina is very small definition of that. So it is basically physical fitness with psychological strength also. So I will discuss in detail afterwards also, but there is a concept called as mental endurance also. So this endurance, generally people do to improve this endurance, many people do exercises. Everybody knows about it. So I just wanted to tell you there are two type of exercises, aerobic and anaerobic. If you see last part of the each point, cycling, swimming, aerobics, rowing and long distance running are aerobic exercises. Whereas sprinting and weightlifting type of exercises, that 100-meter running, these kind of sprints, they are anaerobic exercises. Because what happens is, in first type of exercises, the muscles which are used are different. They are slow moving muscles, whereas in anaerobic they are different. So it is a fast type of muscles. Second main important difference here is in aerobic exercises we need to use oxygen. Then only body will create energy which will provide to us by the overall body processes to make it progressive, and this exercise can be maintained continuously for long time, like marathon runners are using this concept. And most of these exercises are rhythmic. If you see the pictures, images, cycling or this running or rowing, these are all rhythmic exercises. So that is a symbol that it is indication that it is a aerobic exercises. So ultimately oxygen is important for the muscle, whereas the other one are more intense but short duration. They are without oxygen, and these examples are already we discussed. Now, ultimately, what is ideal? Ideally, one should have all these things in rhythm so as to run. So now from here, I am going to focus on aerobic exercises, which will need uh, oxygen to perform. So when you talk about aerobic exercises, ideally we need to improve our cardiovascular system physiology, skeletal muscle physiology, brain and nerve physiology or CNS and ANS, and then metabolic systems to get it more energetic. At the same time, after getting some time of exercise, people will feel a soreness, and that's why they need some rest in between the next level of next cycle of exercise. So that is called as recovery. That period is called as recovery product, recovery period. So ideally, we need all these five. In first, say for example, what we need is we need best locomotion. The body needs to move fast. We need good muscle contraction. We need storage and effective utilization of ATP, which is a form of energy, by the way. And next is the heart should efficiently work, which will transport oxygen. Lungs should work properly. Oxygen should be there. That is also related to oxygen. And blood should carry this oxygen. Say, for example, RBC should be in good health. Muscle, brain is also need to be properly in control. Endocrine system, immune system, all this overall physiology has to be in shape. Then only we will get uh, training, a proper performance, which ultimately goes to endurance. So endurance is a concept where every part of the body is important, and we need something which will improve those things. Now I will take one by one. There are a lot of types of exercises which you already mentioned, 5 types. So metabolic endurance is the first one, where it goes to, this is physiology of the body, which most of us know, so I will not go in detail. It's a basics, anywhere you will find it in books. Ultimately, we are talking about metabolic exercise endurance related to Krebs cycle, energy production from oxygen, which happens in mitochondria, and effective utilization of glycogen to glucose. When there is a need, glycogen converts into glucose and glucose goes to skeletal muscles. So this is a metabolism which is happening. There are certain compounds already there in the research. People found it, uh, and I'm giving references. If you see this pink color, they are basically references which I just listed in that slide itself. So everybody, what I'm going to talk is basically based on evidences. So these are evidences from prior art. I will come to the development of Enducor also, but this is actually at present the normal prior art which we know. So conjugated linoleic acid increases fat oxidation to generate ATP. Normal carbohydrate, which are also required, what we have in our food, which maintains blood glucose metabolism. Creatine increases the synthesis of phosphocreatine and muscle creatine storage improves. If you take proteins and amino acids from food or from supplement, these are the benefits which you already know. And free fatty acid oxidation is main role of protein and amino acids. I'm talking about the role in the metabolism towards the endurance. And BCCA, that is branch chain amino acids, improves the skeletal muscle carnosine content. So it is basically building block of the skeletal muscle. Why it is not going? Okay. Next part is skeletal muscle. So partly it is related to metabolism only, where glucose to convert to glycogen converts to glucose, and then pyruvate cycle it happens, and mitochondria is there are many reactions which gives ultimately energy which is used for high intensity exercise performances. There are certain iron, calcium or phosphorus or number of other ions are also exchanged between the muscles and the external fluids, and then muscle will perform in more efficiently. So this is again a TC and Krebs cycle, which already know that is useful for ATP production. So here nitrates increases efficiency of mitochondrial respiration. This nitrate, these compounds, what I am giving here in bold, they are basically supplements which are already available in the market, which are long back people developed. So creatine is also another supplement which increases ATP synthesis indirectly. So ultimate objective here is increase ATP and energy for the skeletal muscle endurance. The third type of endurance, which is also very, very important, is cardiorespiratory. Again, it depends on oxygen. There is a series of reaction. This actually diagram is what basically is done by trainers to train the athlete for higher endurance, cardiovascular endurance, cardiorespiratory. What they generally do is they go to high altitude so that this athlete's body will be adapted to that low oxygen content. So body is always adapting to some changes and try to compensate it. So these are some compensation body will do, increases plasma volume and increase red cell mass in response to low oxygen content. And then series of reaction takes place, which ultimately body try to increase blood flow to the active muscle and oxygen carrying capacity of lungs and muscles, lungs and heart will increase with this through these reactions. So that increases endurance. So that is what trainers do. In lungs, naturally, this diagram shows different capacities, which we learn in our physiology lectures. Vital capacity is the most important in that, and the another derived factor here is a VO2 max. This is very important physiological parameter which really shows us what endurance levels you have. What is your stamina? In fact, now VO2 max is becoming so important that many people believe that it is a marker for aging. That is now research is going to that level now for VO2 max. Is still under development, but what ultimately in general we can say that increased VO2 max will increase endurance, and increased VO2 max is also indication that lung is in best shape and person can be live for more amount of time. So L-arginine is a one supplement which increases this VO2 max, and apart from that nitric oxide blood flow increases and time to ultimately the the main parameter or a standard. How will you know that increased endurance means exactly what? What physiological measurement is you can do? So ultimately you should do time to exhaustion. If you run, how much time you run until you exhaust? Or if you are cycling, so how much time you are can be pedaling that cycle until you exhaust? So endurance is ultimately in time to exhaustion. Increase is better. Okay. So another type of endurance is mental endurance. Ultimately, there is a category of the products in sports nutrition market. They are called as pre-workout segment. So this pre-workout segment means if you don't feel to exercise, if your mood is not there, a person is physically also cannot do proper exercise. So ultimately endurance time or the time to exhaustion will decrease if the person psychology is not person is not in a psychological status to do that exercise. If the motivation is there, now there are certain characteristic of nervous system which are important for this, and there are definitely neurotransmitter which are related to that. So objective is to increase these neurotransmitter levels for alertness increase, attention, concentration, intuition, increase appetite is another part, of course, that is different area, and satisfaction. So these are the traits of a person which will motivate the person to do exercise, and they will help to do better exercise automatically. So caffeine is a lot very very popular and uh, used product today for the pre-workout segment. But of course, caffeine has a lot of side effects, which everybody knows. As a pharmacy person, people or pharmacy professional, we know this problem. But at present, caffeine is the one which is used. They generally called as caffeine shots, or even there are certain drinks which are there to many sportsmen use those drinks to increase this psychological thrust for the exercise. In fact, there are certain companies which makes $1 billion of business only on drinks. This sports drink, I'm talking about specific sports drink. I'm not talking about the soft drink, which are that is a different segment together. But they are specialized. These drinks are specialized for pre-workout segment. So what ultimately they do is they increase the neurotransmitter levels. And second possibility, what they generally do, there are many compounds, particularly vitamin C do it, is decreases cortisol. And cortisol, we know that if you can decrease that, it will decrease mental fatigueness and increase endurance. There are direct references which can link this thing. If you see these references, you will get exactly those studies, or sometimes there are some consensus statements which are made by some very reputed sports nutrition or exercise physiology organizations, and some of the good journals in that. So you can go through it in details for those specific relationships. And the last part, which is so first what we saw, mental endurance or cardiorespiratory endurance or metabolic endurance, we should try to increase that. The another thing, this muscle soreness and recovery, these are the places, these are the limitations. Body will stop sometime. Body will show that it will now it cannot go ahead. Exhaustion. That is we can call it as in normal word. We exhaust. So exhaust means basically what happened here is once you have started training, there is a low grade inflammation comes in our body, in a muscle. This is generally happens in muscle. And this because of this, for example, NF kappa B accumulation, the muscle soreness is there. The another part of that is a lactic acid accumulation. So that is also, now here I showed the two different processes, which is a oxygen utilization of oxygen and utilization of nitrogen. So in one process, it is a lactate utilization, and at the same time, body is doing lactate accumulation also. So in when you have reached to some limit, lactate will high accumulation of lactate is there, and that's why body will not mitochondria cannot now produce more ATP molecule, that is energy, and that's why a person has to stop there. So there are L-arginine like compounds or creatine like compounds. They say, for example, creatine decreases inflammatory cytokine, or curcumin also. Curcumin is very well-known molecule. We know that. And lactate accumulation also can be decreased by L-arginine like molecules. So if you take this type of supplements, you can get lower muscle soreness and increases muscle recovery. Muscle recovery improvement means you can stop for less amount of time for next level of exercise, something like that. And for the endurance, athlete want to do more and more exercise without having muscle soreness or recover. They need faster recovery. So this kind of product, L-arginine or creatine, these kind of products, they are categorized under recovery products, muscle recovery products. So they, if you see the market, they are sold under different category, whereas caffeine-like compounds, they are sold under pre-workout category. So different products in commercially when they are selling, they are under different category because of different physiology, because of different claims. Now, the major, if you really see for the athlete, he has to take one pre-exercise, pre-recovery, pre-exercise product. Then he has to do something else to improve cardiovascular respiration, endurance, and then he has to take some product for reducing muscle soreness. So this thing will happen. So ideally, what we want is one product or one at least composition, preferably natural product, can do this. And that is what exactly we tried to do with, with the raw material fenugreek, which we work since long time. So these are all details which we already everybody knows about fenugreek, Trigonella phanegraecum, scientific name, particularly abundant in India, and but also there in Middle East and other many countries, Southeast Asia, also some places. A long history of use, everybody knows fenugreek. This is also generally recognized as safe, very, very important characteristic for any product to go into the sports nutrition market or dietary supplement market, because yeah, that will increase the chances of applications. And recommended daily dose 25 gram is basically decided based on sugar level things, not related to sport nutrition. And this 25 gram is a recommended, it is not highest. And even if it is recommended, 25 gram of this is RDI is for fenugreek seeds. I am not talking about the product, fenugreek seeds. And naturally, 25 gram of seeds nobody can eat at a time. Uh, just like powder, if you do powder, it is very bitter, first of all. And some problems can arise because it has very, very high content of fiber, very high fiber compounds. Compound is there, fenugreek seeds, a lot of fiber contents are there. So we are isolated major bioactives, glycoside, amino acid, alkaloid, and there are a lot of patents and many other products we already have. Here we are just talking that because we know some of the products, some of the markers from these products are beneficial for sports nutrition. Specifically, we believe that the concept which we just discussed before this slide, this claims can be made because there are certain properties, but we need to study it. So what was already known for us was this: fenugreek seed extracts, standardized extracts with glycoside marker, which we knew from since long that it is having exercise performance, muscle performance is increasing. But we are talking more about on the strength part, not on the endurance part in this paper. These are our papers, of course, Pool et al. This is published by us, Indus Biotech. And then another paper which we did in 2022 was for testosterone boosting. We have one product which is quite popular in US and all over world. It's actually leading product in Amazon in US in this segment, which is testosterone booster. So this is another one paper which we have. It is a review basically, and there are a lot of other papers which are supporting in this review. Taylor et al. Is related to one paper where we added our fenugreek extract, which is rich in 4-hydroxyisoleucine, which I will come to that, which is amino acid and trigonelline. So this is we found useful for creatine delivery. Then another paper, this paper is not from us. It is published in 2018 by some other, I think they are from Egyptian or some place, Turkey, I think. So they did this clinical trial on endurance exercise athlete. But if you see, this is fenugreek seed supplementation where they haven't mentioned any marker. Second thing is they are just giving powder of fenugreek seed, which are given in the huge amounts. So this paper was there, which gives some idea, but that was not directly focused on endurance. It is only focused on respiratory endurance, VO2 max, and fat mass is there, but not fat metabolism. So this was one paper which was not published by us, but some other we got, and some few papers, more papers also were there, which are of less importance and not clinical. So what we got now, we are talking about our products and our science based on 4HI. This is our molecule which, see, this Endicor which I am talking about is having three marker compounds. One is 4-hydroxyisoleucine, second is trigonelline, and third is some selected glycoside. Sorry. So these three, one by one, I will tell you in each slide the prior art related to those compound, but which can be useful for our direct endurance enhancement activities. So if you see the last column, which is more important, where 4HI increases endurance capacity, there is a papers in this. Time to exertion, blood lactate concentration will decrease, post exercise glycogen resynthesis will increase, serum cortisol level will decrease. So if you see all these properties which are helpful to improve this endurance, cardiovascular endurance, muscle recovery, enhancement can be there, metabolic endurance. So then second compound also we have some papers. Some of them of papers are we published in the past. Some of them are others papers. Like in mental endurance, Gaur et al. Is our paper, whereas Mirazi was some other some institute they published. So ultimately, trigonelline is another marker which we can use for mental, metabolic, and endurance enhancement. Mental and metabolic. Sorry. With this, these are certain mechanisms by which it will do it. And third marker was a glycoside. Again, same thing. These are actually all the four papers are our papers. There are one animal study and 3 clinical studies. So most of the studies which actually, if you see, they are improving the skeletal muscle. They work on skeletal muscle to reduce the muscle inflammation and increase the muscle strength. So all these papers, if you see, combinedly or together, we can say that, okay, if we can have composition with these 3 markers with a proper quality, and we can get a comprehensive endurance solution. Comprehensive endurance means in one compound should be able to do all 4 type of endurances. An athlete can take only that particular thing and go away. There is no need of taking A, B, C, D, E for that. Naturally, cost will also decrease. So this is what we thought. We will do standardized composition of fenugreek seed extract with our, of course, there is a technology behind it, how to extract and how to make those things. But ultimately, we have to standardize on these 3 markers. So we could standardize up to 12% of each, because the composition has to be proper. So 12% each we could achieve. Then what we did is, now when we achieve this, it was not done by single extract. So we have to do 2 different extracts and mix in a proper proportion to make this 12% each. And both these, I'll say, extracts individually was tested for this toxicology battery of test. If you see, these are four references, because they are derived, one for one compound, a second one, two papers are for one compound and 2 papers for second compound. These papers are having all the information. So these 2 extracts which we mix together to proper proportion, we got the Endicor with 3 markers. So on that, then we did animal study for efficacy testing and mechanism. And second study which we did is a clinical trial, which is a double-blind placebo-controlled clinical trial in specific endurance athletes. First study, animal study, we did in India. Second study we did in Australia. And if you see, first study uses actually treadmill running exercise. So Pune College of Pharmacy had this instrument specific for the laboratory rats. And under supervision of Arul Mozi madam, we did this. So I'll explain these studies, both of the studies with you. So if you see the diagram, the right hand side, this structure with five rows, this is basically treadmill, animal treadmill. Naturally, it will have some constraints to go, that animal should not go outside. And these are actually treadmills which are having belts and they are running. So this kind of thing which we did, these are the dosing which generally we pharmacologists do with multi-dosing studies. Once daily, 28 day was we now this one we got on the basis of previous papers, how much we should be should be able to test. So here what we measured was a time to exhaustion. So this was the instrument details. Metabolic parameter also we measured: glucose, protein, fat. Then glycogen levels in gastrocnemius muscle we measured. Cardiovascular parameter we measured with the power lab, which is known instrument. And respiratory parameter also we measured with a whole body plethysmograph. This was also there. This is also there in Pune College of Pharmacy. So basically, the instrument combination was made us to do this possible this study. This images I took from their sites, like Vijay Instrument and MCA Technology. These are not directly photographs of the instrument. This is Power Lab, 8-channel Power Lab. So now, if you see the result, very clearly I can show you wherever there is a green color, they are beneficial effects. Okay, there is a gray color, there is a neutral, like I will say. So like glucose levels decreases, gastrocnemius muscle, their glycogen levels increased, which shows the metabolic endurance improves. So likewise, so here I tried to show with the same figure. If you remember those same figure I am putting here, but now instead of those other compounds, I am trying to tell you how 4-HI might be useful to create this improvement in metabolic endurance, which we saw in the last slide. So what is happening is 4-HI is increasing post-exercise muscle glycogen resynthesis, which is references are there, and which increases carbohydrate and glucose transports into the skeletal muscle. In fact, this second one, people says that this is the reason why 4-hydroxyisoleucine is giving anti-diabetic effects or anti-hyperglycemic, I will say. This is known references for that. And that this glucose transport is useful to increase endurance also at the same time. If the person is diabetic, it will give anti-diabetic effect. If the person is normal, it will increase his endurance capacity if the person is trying to do aerobic exercises. And fenugreek extracts, also there is one paper which is reported as a general increased glycogen preservation. Of course, they are not directly talking about 4-HI here. It is a general fenugreek extract. The second result what we got is on cardiovascular endurance, where all ECG parameter and all arterial parameter like heart rate, BP, these are all favorably showed differences. Now here, I'm talking about non-significant and still I'm keeping gray because this is actually systolic BP should not decrease significantly. Okay, so that is important. Now here you will see. You might be knowing that many of the athlete or runners, they die on the treadmill, or many people who try to do, there are a lot of deaths happens on the treadmill when they do exercise. Generally it should not happen because they are doing exercises, and exercise should prevent the person from death, should not cause death. The problem here is they are having this BP, systemic BP, LVESP and LVEDP. See, LVESP and LVEDP is left ventricular end systolic pressure and left ventricular end diastolic pressure. These three parameters should remain unchanged at the time of exercise. Now, major issue with many supplement is they increase this, and that will give a problem with the cardiac safety profile is a danger. So supplements taking, we have to be very careful unless people know. For example, caffeine is increasing this. So if somebody is taking too much caffeine, and caffeine based supplement also, of course, caffeine shot, generally people take caffeine shot, very small, very strong coffee they take before exercises. So those kind of things, why they are not desirable is because of this. So this is one of the major safety feature for this product which we thought, and that's why we marketed. Otherwise this was our major focus that time, whether it is happening. So this is in animal study, by the way. We have to remember it has limited uh, reliability, not reliability, but limited conclusion we should do with that, unless we do clinical trial, which will come to that. I will come to that. So otherwise also their QTC interval is also very important parameter, and it is prevention of HRV also reduced. So QTC is also another important parameter where cardiac safety and efficacy is shown clearly by this. So that this is a major slide which where I can say that this is a good product to start with. Similarly, respiratory parameters are also very positive. So cardiovascular and cardiology and respiratory parameters we found extremely positive with very good safety. Here also you can see a lot of clear advantages, clear significance of that with the particularly dP by dt and all these parameters, end diastolic volume, everything is perfect, very good. We got ejection fraction is another very important parameter. If your ejection fraction is not increased, endurance is not going to happen. And if other things are positive but ejection fraction is not good, problems will be there. Respiratory also very nicely we can sort, particularly all the volumes are in positive side or benefits. So here what must be happening with the Enducor is basically increases time to exhaustion, and which we sorry, this is repeated. I have to delete it. And in this Enducor is showing VO2 max increase. It might show oxygen saturation and lung function increase, tidal volume increase, and decrease pain. So this is what a mechanism which is might be happening here. Similarly, skeletal muscle soreness. By the way, one of the component was already shown in clinical trial that respiratory, inflammatory markers in skeletal muscle decreases in endurance athlete, which was already known from before that they are glycosides. But when we give this Enducor, they showed very three characteristic markers where it should decrease actually. So beneficial, significant benefits are shown here. The stars, we already know that they are statistical significance, by the way. So direction is important. Wherever I showed the green, they are beneficial, significant benefits we got. So creatine kinase, lactate dehydrogenase, and BUN. These are actually markers which people are using in clinical trial also. They are benefit. Enducor showed significant benefits. Then in IL-6 and IL-1 alpha reduction in the levels are there. Pre-workout and post-workout levels are shown here. This is basically a result of clinical study. This is not a animal study which I showed you. This is already published clinical study which we did with one Baylor University in USA, and that we got. This is double-blind placebo-controlled study where we took 400 milligram once a day for 14 days in 20 non-resistant trained subjects, and these are exercises we have to perform as per protocols. And then we got significant decrease in IL-1 and IL-6, which are anti-inflammatory effects are seen here. So whenever there is a anti-inflammatory effect, muscle sore, particularly in muscle. So muscle soreness will decrease. This is what is happening. So these are the mechanistic figure which I showed earlier, and now I am showing Endicor how what may be the proper mechanism to or possible mechanism to decrease the skeletal muscle soreness. Like we already discussed, inflammatory markers are reduced, but 4-HI and glycoside extract is already shown this pro-inflammatory cytokine reduction effects. This is in vitro study which we did, and references are there which we published also afterwards. Here, second possibility is because Endicor showed decrease creatine kinase and LDH. That is a that is a mechanism by which it is decreasing the lactic acid also, and to decrease muscle soreness and increase muscle recovery. So this is another possible mechanism for skeletal muscle soreness. One of the marker of 4-HI and trigonelin, which I told you there are 3 markers, out of which 2 marker, that combination which we already tested in vitro in one of the experiment in the past, copper ascorbate induced oxidative stress to mitochondria, and we took organs like heart and lungs, etc. So here I'm talking about goat aortic tissue was taken, and the effect on the enzymes on TCI Krebs cycle for ATP generation was studied. So if you see this all four enzymes which are involved are showing beneficial effects. If you see two enzymes are needs, actually it is a positive side, it should decrease, and these two enzymes should increase to give better um, effects on transport chains and ATP production. And this actually we did in heart mitochondria after giving oxidative stress. Now, actually, what happened is high intensity exercise is itself is considered as oxidative stress, and that shows the typical effects of oxidative stress in the person. And one of the symptom of that is TCA cycle get disturbed and ATP production decreases. So here this 4-hydroxyisoleucine and trigonelin is showing effect in that direction. So this is what I actually told in the form of figure. So Endicor with these two markers actually increases Krebs cycle and electron transport chain enzymes, improves ATP synthesis in vital organs. Actually, I just showed the effects on heart, but actually that experiment we did with lung tissue, skeletal muscle we did afterwards. But in the same experiment, lung was also tested. So muscle glycogen resynthesis and lactic accumulation decreases. So this is another. These are the mechanism of 4-HI and trigonelin towards improvement of ATP and energy. Ultimately, it will increase the skeletal muscle endurance. So now these slides were related to in vitro or animal studies. Now I'll show you after that studies when we are convinced that yes, it can be good product to test in clinic. So we did actually clinical trial. These are the details actually. 8-week treadmill exercise. We, athlete, we are trained male subject, of course, because of the in the hormone interference. We tested this. We did this clinical study in male subject only. This was done in Australia in one of the center in Perth recently. I think 3 years back. 3 years back. So we did a placebo, and then we tried to use two doses, 300 and 600, because at that time we were not sure which dose will be effective, or should we go to 300 only. So a 3-arm study, 2 arms of active and 1 arm of placebo. So these are the major parameters: time to exhaustion, cardiorespiratory, metabolic, and this endurance related parameter, which I will show you in next slides. Now what happened is both the studies are actually, we studied parameters on endurance. And in both the studies, we tried cardiovascular endurance, metabolic endurance, and mental endurance. Only point was many of the parameter which we could test in animal study, which we couldn't do in clinical study naturally. Because in animal, you can remove the muscle and measure glycogen. In human study, we cannot do it. So, but ultimately, to make the claim, human studies must support the animal study. So if you see, these are the experimental protocol. One more point I wanted to stress here is when we do any sports nutrition study or exercise physiology clinical trial, we have to follow some international guidelines on exercise schedules or exercise, you can say, protocols. And in a paper, we have to mention which one you have used, because based on that, the athlete decides, okay, this modified Balke protocol is basically used for by endurance athlete or cyclist or swimmers. So these are typical people who use these protocols for their training. So we use that's a relevant protocol. That is very important. And this protocol uses treadmill. Although it is treadmill, it will be useful for many type of athlete. It is not restricted to the people who are just going to gyms. That is what I wanted to tell, the relationship of this protocol. So these are some schedules. 5 minutes you have to rest, then walk for 3 minutes. Gradient should be 1%. These are all things are there. We have to will not go into detail. These references are there for the details. And then after 30 minutes of this protocol, we have to do wind gate test. This is based on cycle ergometer. So first treadmill, then cycle with some time in between, 30-minute rest. So this is basically protocol which is generally followed by, this is internationally accepted or standardized protocol. So we followed that same. So these are some of the parameters we measured and we got some good effects. Apart from that, we measured many other parameters. So where we haven't got any effect, but thing is, we haven't got any bad effect also. So that was all the results were in the physiological ranges. It is not exceedingly high or exceedingly low. But the other effects were not significant. Whatever is significant, that only I am showing here. So what happened here is time to exhaustion will increases by 5%. Now 5% is a good figure here, considered to be very good. If you consider the extreme protocol which we just followed, then total distance running, naturally it should increase. So about 6.5% distance they could run more. Run more means that is on cycle ergometer. It was not actual running. On the ergometer, in this center, which we have done in one of the good center in Australia, which are meant for to test this, they have all the measurement, I'll say, mm, uh, probes and sensors, and that is connected to the machine. Actually, I should have put that photo here, but okay. So that those machines are giving these parameters, and it is done 3 times in between also, because for zero baseline, 4 week and 8 week, all the people are doing this by coming to that center. And in between, they are exercising as per their schedule. They are also have a standard schedule told by the people. Of course, we have not maintained their food profile, which we couldn't do. So then another parameter which we did is a metabolic equivalent. So higher is better. Here also we got 7% increase. And another parameter which we got particularly with respect to non-esterified fatty acid, free fatty acid. So this should decrease. So 38% decrease is there. So what happens here in endurance or any sports, body will take some time to adapt to the new situation. When body is doing adaptation in exercise, body will also take time to show the effects of supplement also. So here we took 8 week. Maybe it is showing in 3 to 4 parameters. Maybe with the increase in time, we might have got more effects. But only thing is, at present we have to focus on those effects which we got significant, and we are claiming only those. So here what is happening here? is, see, 1 MET is basically amount of energy used while sitting quietly. So endurance increases this MET, which is indicator of increase of endurance. Second thing is decrease of NEFA in blood shows increase in this utilization in muscles. This NEFA is utilized to increase the ATP production. And that is why direct ATP we cannot measure. We couldn't measure. But in animal study we measured. So if you that is the advantage of clinical and animal. These are two different type of experiment, and we have to see which one and which one has some limit and try to overcome those limit in other type of experiment. Doing two animal experiment can give more information, but the limitation would have been this only. Then endocore will decrease NEFA. We saw that, and we believe that 4HI is the responsible parameter, because this 4HI is known to decrease this NEFA from previous relationship slides we can see, and that might be increasing in swimming endurance. This is reported paper. Decreases NEFA. Decreases NEFA is not reported, but increased swimming endurance is reported through NEFA. This relationship is reported in this paper number 3. If you see, actually, these papers are not very new. Some of them are very old, because the basic papers are already done. But now what we try to do here is, what is the basic science, what is the evidence, and then do the test, what we will get evidence, and correlate those evidences, our evidences with previous evidences, see the whether they are correlating. Sometimes it may opposite also, but we couldn't find it in this case, opposing effects at least. And whatever we got in clinical trial, they are supported by either our animal studies or the previously reported animal studies. But this is the first study on fenugreek, any fenugreek extract for endurance, which barring which is standardized, because the previous one was not standardized compound. We don't know what exactly in that. Even though although it gives some direction that yes, we got some confidence. Yes, if we do, we may get something. Another very important result, this is most important, and which we got support from animal study also directly, is VO2 max. As I told you, VO2 max is very important parameter, which shows maximal oxygen consumption. Naturally, in aerobic exercises, this is the most important. So in this study, we got these numbers, some 5%, 3%, about 4.5%, about 3%. So this shows overall, particularly VO2 max, I will say that this is most important parameter which indicates a direct indicator of endurance increase, which is basically related to cardiovascular system to deliver oxygen to the exercising muscle. And this relationship, VO2 max, is related to this. This relationship is shown in this one, which is very old paper, which is established. See, actually many of the things are established relationships here. So VO2 max with endurance relationship is very much established. Now next part is mental. Now in our study, we haven't got much, we haven't got opportunity actually to study mental endurance parameters. Very small thing which we couldn't do in clinical study, neither we could do in animal study, but we have number of other prior art proofs where trigonelline is known to increase dopamine, and 4HI is known to increase serotonin, which are some of them are our paper, and I think one is not ours, but they are very established facts there. So we can say these markers are there, and that's why they might be improving this mental endurance, and cortisol decrease. So that is also there. So apart from mental endurance, which we couldn't get direct proofs from clinical study, maybe we will do next study, we will definitely try to increase. So we can say at the end, clinical evidence is suggesting. Now this this is a paper actually which we did conference presentation. And the study which the paper which I showed in the earlier slide was a public actual journal publication of the same paper. So here I just wrote Mills, but actually there are one conference presentation and one journal publication we did on this. So here we got 600 milligram daily for 8 weeks with the treadmill exercise in recreationally trained male. Recreationally trained male subject means they are not first time they are not going to gym. They know they are actually normal gym goers, but they are not aiming for any sports or sports extremes. Like they are not trying to participate with any state competition or anything else. So they are just people like you and me. They are trying to become fit and try to increase the endurance. So safe and well tolerated, no issue on that. Time to exhaustion increases. Total distance run was increased. NEFA decreases, MATE increases, and VO2 max. This all respiratory parameter increases. So respiratory endurance, metabolic endurance, all increase. So these are the now see. This is a scientific wording, and this is a market wording. Enhances metabolic endurance, enhances cardiac endurance. These are. This is just wanted to show science and commerce connection. So at the end, we try to show what is a mechanism of action. Most of this is I already mentioned in the past slides. Here I wanted to tell you some metaphor. See, assume that skeletal muscle is a furnace. So to have furnace and get some, now this is a very cold season also, and I'm sure Nashik will be more cold than Pune. So everybody is trying to get some warmth, some heat. So furnace is one of the things which people do in their home sometimes. Not in extreme cold, they generally do in maybe Himalayan areas. So this furnace, to get that furnace run, what you need? You need fuel. So and I'm just thinking that exercise, endurance is a furnace-like thing. Naturally, because of that only we get heat after exercising. So we need fuel in terms of glucose, fuel in terms of oxygen. So one type of fuel is given by muscles, which is a top portion. Second type of fuel is provided by heart and lungs, which is the right side, with increased VO2 max and increased cardiovascular performances. And this fuel will also need the trigger. So unless you have some trigger, if with some flame, it will need some flame to start. So if the starting flame is not there, it cannot run. So our mental alertness, concentration, or decreased fatigueness or energetic thing, which is provided by brain neurotransmitters. And together, if this flame doesn't have continuous oxygen or continuous things, it will go away faster. So you need to provide continuous supply of oxygen, and that is basically given by ATP levels, and it should not have any collection of you know uh, some dust or any other things in it. It should not have contaminated. Wood should be pure, or so those things are actually acting as a in our body. Pro-inflammatory cytokines are doing that. It will not let skeletal muscle to run to the up to that capacity. It will show muscle soreness. So this is just trying to metaphor, and this part I am just trying to give. It is already in market. You can go to Enducore website and see these details, what I just told. I just wanted to focus your attention on this USP. Can you see all of them USP? Other things are we already discussed. So whenever you go to market, we have to come out with some special things. So here you can see non-GMO product, ETO free, 100% traceability to raw material. These are something which are more or less administrative issues, or not purely administrative. We have to do lot of other testing other than clinical trial and preclinical studies, and they are generally done by our quality assurance people. quality control people. And on the right hand side, if you see certifications, it is a result of that. This is done by our regulatory affair people. So this is a teamwork. What we showed is a teamwork which has, we have team of scientific affairs, regulatory, quality assurance and quality control. This is a combined result of that in last few years. So this is, you'll find it on our website. Certifications are necessary to build a trust from the consumers that yes, there is some quality in this product. Now I will try to run one video which is there on our website. You can see that link also. But let us see. Can you once it is run, one of you can talk that whether it is sound is coming to you, just to test.
Endurance is an ability to
Yes,
sustain
sir.
physical activity
Okay. Yeah.
or mental efforts for a longer period of time. Endurance is influenced by factors such as energy production, cardiac output, oxygen utilization, mental drive, and muscle recovery. Enducor. Enducor is an innovative fenugreek seed extract with proven clinical benefits in enhancing exercise performance through improved endurance. In a double-blind, placebo-controlled clinical study, innovative fenugreek seed extract, Enducor, exhibited a 5% increase in time to exhaustion and 6.53% enhancement in total distance covered in a treadmill exercise. Enducor achieves its effects through optimized parameters, enhanced cardiac function, improved respiratory efficiency, elevated energy production, increased mental motivation, minimizes muscle soreness. Enducor improves cardiorespiratory capacity. Engaging in physical activity or exercise escalates the requirement for oxygen and nutrients within muscle cells and throughout the body. Enducor enhances the distribution of oxygen and nutrients to various body regions by notably augmenting cardiac output by increasing ejection fraction. According to findings from an animal study, Enducor displayed a 24.32% elevation in ejection fraction, signifying its potential to enhance circulatory efficiency and nutrient delivery. Respiratory function. The greater the oxygen utilization during intense workouts, the higher the energy generation, enabling extended maintenance of physical effort. A person's aerobic capacity, denoted as VO2 max, reflects the peak rate at which the heart, lungs, and muscles can effectively utilize oxygen throughout exercise. Enducor exhibited a 5.29% enhancement in VO2 max, highlighting its potential to elevate oxygen utilization and consequently energy production during physical activity. Improves muscle glycogen storage. Cellular respiration encompasses a series of metabolic processes within cells that convert acquired biochemical energy from food into adenosine triphosphate, ATP, a vital cellular energy compound. ATP serves as the cellular currency for energy exchange, with oxygen playing a crucial role in this process. When oxygen is present, cells can convert a single glucose molecule into 38 ATPs through the citric acid pathway. In the absence of oxygen, cells adopt the lactic acid pathway, producing 2 ATPs. Oxygen's significance extends to fat oxidation and ATP generation from fats. Enducor enhances oxygen delivery to cells during exercise, achieved by boosting cardiac output and VO2 max. This improvement facilitates two key benefits: efficient generation of more ATPs from limited glucose, delaying glycogen depletion, and enabling the oxidation of free fatty acids to meet escalated energy demands during exercise. Endicor demonstrated a 38.28% reduction in plasma non-esterified fatty acids, signifying more fat oxidation, alongside a 25% rise in muscle glycogen storage levels. No soreness and pro-inflammatory markers. Physical exercise prompts the body to release pro-inflammatory markers like cytokines, contributing to fatigue and muscle soreness. Endicor mitigates this effect by decreasing pro-inflammatory cytokines, leading to a reduction in muscle fatigue. This is achieved through a substantial 51.87% decrease in LDH serum levels and 7.03% reduction in BUN levels. Mental motivation. Serotonin and dopamine levels. Dopamine and serotonin, often referred to as the happy hormones, play a pivotal role in modulating mood and driving motivation for both physical and mental endeavors. Endicor demonstrated a substantial rise in serotonin, 5-HT, and dopamine levels within the brain, showcasing increase of 77.88% and 60.08% respectively, as compared to the control group's brain serotonin, 5-HT, and brain dopamine, DA levels. Endicor also shown 34.09% increase in brain ATP synthase. Unlock your peak potential, Endicor.
So this video is also there on the YouTube channel of Indus Biotech. So here I wanted to show you now last 2-3 slide. I wanted to show you how our scientific knowledge can be very useful for any product development and naturally for a company, any company to develop the proper product which will be useful to the people, to the segment, which we generally should be identified beforehand. And there are certain marketing things they can do uh, uh, because this multimedia video presentation video was made by some other people, like those who know about video production and audio, this kind of thing. But the content what we generated through projects or publications and even prior art is also very useful. Uh, from that only we have to collect the evidences and we have to base our hypothesis and results, conclusions only based on evidences. So I would like to say that with every time evidence based uh, research should be done. So this is all about from my side. I think if there is any questions, I will be happy to answer that.
Thank you, sir. The session is open for question answer.
So before that, I would like to acknowledge the contribution that SNJB Pharmacon is happening now every year, and it is doing good discussions and good topics are being discussed in depth. And this year it was online. That is also very nicely organized. So I would like to give thanks specifically Dr. Upasani and the whole team of SNGB Pharmacon. So thank you.
Thank you, sir. Now, sir, explain various types of endurance, such as cardiovascular endurance, respiratory endurance, metabolic, mental endurance. He also focused on the importance of preclinical and clinical development and reconstruction and advancement of Endicor product. Sir, really give insight on the development of this Endicor product. Again, he also explained the different mechanism of action, how the different developments or endurance levels that are increased by using phenolic. Sir, really from SNJB Pharmacon, and from our team of Pharmacon, we are really thankful for you for giving such a insightful for this development of Endicor product. Now we come to the conclusion of first day of SNJB
Excuse
Pharmacon.
me, madam.
Yes, sir.
I have one question with Thakur Desai, sir. Sir,
Yeah, please go ahead.
how will you manage the dose of this product, Enducare, in case of human being?
It is a clinical trial, na? 600 milligram once a day.
600 milligram once a day. Okay.
See, this product basically, I forgot to tell you, this is not a B2C product at present available in market.
Okay. It is
So,
under trial.
no, not trial. Clinical trial is done.
Okay.
So what we are doing is we are selling this product as a B2B product.
Okay.
This is not now. Last year I gave lecture on Fenuflex, which was available in market in Amazon.
Right.
So this product is not directly available to the consumer. So
Okay.
we are discussing with various people, business people in
Okay.
India as well as other places. So they will have some product with some other name. Basically, Enducor will not be name of the product. It may be, I don't know, but that is a different commercial discussion.
Okay.
Enducor is a branded ingredient. So I wanted to tell you concept of branded ingredient. So this
Okay.
is not a final product.
Right. Thank you.
Your
Prasad,
website.
sir,
Yes.
just a minute, sir. Uh, just out of curiosity, I got a question. We generally we Indians do consume this fenugreek during winter season to beat the cold. Have you come across any mild side effects or mild due to the increase in acidity or acid reflux by the by consumption of these
I
products?
don't
I mean,
know
fenugreek.
this cold application. People generally use for their sugar levels, but they do it with soaking of the seeds in the water
Yeah.
and
Yep.
taking it in the morning. So that will be very less dose. Actually, according to me, it will not give any good effect also. Forget about harm effect, bad effects, because they are in extract or very small quantity they will consume. You are asking about maybe direct consumption of uh, powder,
Seed.
seeds. So how much we are talking here? Because every toxic effect or every side effects depend on exposure. Exposure in terms of dose, exposure in terms of time. So it
Okay.
depends on that. So here in this case, what we did, uh, there are general. See, we are scientists, so we cannot talk general things, because when we talk fenugreek, we need to talk about marker. We need to talk about standardized things, standardized extract. So very difficult to tell exactly what will happen with exactly how much
Yeah, I
amount.
was interested in the other effects which the product is exerting over the volunteers of clinical trial.
So at present, what we did is for 8 week only. Okay. So 8 week, we should not give any problem. It is not actually reported anything in athlete. See, clinical trial is a controlled experiment. You cannot judge everything with the clinical trial. Otherwise, there will not be problems. Now, if you must be knowing, Ozempic is coming to India, but already there are certain problems are already reported. Mental problems, particularly suicidal thoughts. There are certain points are already people are talking, but that is so it is a research. So these things will come out. Although up to 600 we are confident because we tested it. One more point I will tell you. This particular extract, sorry, composition, I told that it is a combination of two extracts. Both these extracts are already there since long time. We are using it, or even there are products out of it from these two extracts. In fact, one thing I told you, that one extract with glycosides, it is there in the US for about 20 years, it is still there. And people are using it. They are using it. We are recommended dose is 600 milligram only, but only glycoside. Here we are talking about combination in total glycoside. So we are quite sure about the side effect, but of course things are research. So whatever is available, based on that we can talk on this extract or this composition. About general fenugreek will be difficult to comment. There are a lot of variations.
Okay. Thank you, sir.
Thank you, sir. Now we are moving towards concluding session of first day of SNJB Pharmacon. I feel both proud and gratified to reflect upon the enriching academic journey we have experienced today. SNJB Pharmacon, which began in its journey in 2017, was conceptualized with a clear and purposeful aim to promote academic research, encourage meaningful knowledge sharing, and create a strong and sustainable research culture in pharmaceutical science. Today, as we successfully complete the first day of 9th edition, we can confidently say that this version of SNJB Pharmacon 2025 has continuously strengthened year after year. The inaugural session of SNJB Pharmacon 2025 set a strong academic tone. The conference was formally inaugurated with warm and insightful welcome address by Dr. C.D. Upasani, Principal, SSDJ College of Pharmacy. His words emphasize the importance of academic excellence, ethical responsibility, and lifelong learning, inspiring all of us to engage wholeheartedly in the proceeding of this conference. This was followed by the preamble delivered by Dr. G.D. Basarkar, the coordinator of Pharmacon 2025, who clearly outlined the objectives, theme, academic relevance of this year's conference. His efforts and leadership have been central to organizing this prestigious event, and we sincerely appreciate his dedication. We are truly honored to have Dr. Saranjit Singh, former director, NIPER Chandigarh, as a chief guest of SNJB Pharmacon 2025. It is a matter of immense pride for us that Dr. Singh has served as an ambassador of Pharmacon since 2017, supporting and guiding conference from its very beginning. His continued association has significantly contributed to the academic structure and credibility of SNJB Pharmacon. Today's first session of day was delivered by Dr. Saranjit Singh on the highly relevant topic stability testing, importance, and regulatory consideration. His lecture strongly reinforced the principle that patient safety must always remain on the priority in pharmaceutical development. He discussed recent FDA warning letters, current research article, real-life case studies of drug recalls due to stability failure, and ICH guidelines for stability, which provided deep regulatory insight. A key takeaway from his lecture was the reminder that even product developed under a controlled laboratory condition can fail its transportation, shipment, and storage environment if not properly maintained. He also highlighted the importance of stress testing of pharmaceutical product as per ICH guidelines. The second session of the day was conducted by Dr. C.R. Patil, Professor R.C. Patel, Institute of Pharmaceutical Education and Research, Shirpur, on the thoughtful topic, Artificial Intelligence in Pharmacy. Is it helping the process of drug discovery to transform? Dr. Patil explained how artificial intelligence is revolutionized in drug discovery, from target identification and lead optimization to predictive modeling and decision making. His session also offered valuable insight into how emerging technologies are reshaping pharmaceutical research and emphasized the need for future pharmacists and researchers to adapt this rapidly evolving digital landscape. The third session was delivered by Dr. Prasad Thakur Desai, Chief Scientific Officer and Vice President, Indus Biotech Limited, Pune, on the topic Fueling with Fenugreek, Compressing Endurance Enhancement with Endocor. He highlighted the strong scientific rationale behind the development of Endocor, a standardized fenugreek seed extract. The lecture summarized evidence from the efficacy, safety, and randomized controlled clinical study. These studies demonstrated measurable improvement in the endurance of heart, lungs, brain, and skeletal muscle. Thus, the first day of SNJB Pharmacon 2025 successfully covered a wide academic spectrum from the regulatory science and patient safety to artificial intelligence in drug discovery and evidence-based nutraceutical research. This truly reflects the core philosophy of SNJB Pharmacon to offer a knowledge feast through interaction with highly experienced, research-oriented, and inspiring resource person. The conference is scheduled over 2 days, and tomorrow we look forward to more enriching sessions by renowned experts. Dr. Prashant Murumkar, Assistant Professor, the Maharaja Sayaji Rao University of Baroda, and Dr. Vishal Zamre, Associate Professor and Head, Department of Pharmaceutical Chemistry, PCET School of Pharmacy, Pimpri Chandwad University. The afternoon session tomorrow will feature scientific oral presentation, providing a platform for young researchers and students to showcase their research and ideas. Before concluding, I would like to express my sincere gratitude to all our distinguished speakers, delegates, organizing committee members, faculty coordinators, and technical teams for their tireless efforts and enthusiastic participation, which made day one of SNJB Pharmacon 2025 a great success. With these words, I formally conclude the first day of SNJB Pharmacon 2025 and invite all of you to join us tomorrow with the same enthusiasm, curiosity, and academic spirit. Thank you all and have a pleasant evening. Thank you once again. I request technical team.
Okay, thank you. Thank you very much. Tomorrow.
Thank you, Prasad.
Bye-bye.
Thank you. Thank you.